হ্যাঁ হ্যাঁ
Hello, good morning.
স্যার গুড মর্নিং।
গুড মর্নিং স্যার।
কথা শোনা যাচ্ছে তাহলে?
স্যার শোনা যাচ্ছে?
হ্যাঁ হ্যাঁ শোনা যাচ্ছে, ভালো শোনা যাচ্ছে।
থ্যাংক ইউ।
Hello, Dr. Anjulika.
Yes, sir, you are audible. Good morning, sir.
Uh,
So I start?
Yeah, please start. Please do.
Okay, sir. So very good morning to all of you. I, Dr. Anjulika Roy, on behalf of organizing committee, welcoming all of you in this national seminar on cytogenetics, in vitro culture, and phytochemistry of plant and microbes for sustainable use, which is jointly organized by Department of Botany, Visva-Bharati, and Department of Botany, Raidighi College. We are fortunate enough that we are able to organize this national seminar with the kind permission of university authority in this prevailing pandemic situation. This pandemic realizes us the importance and need of more research in the field of biological sciences. We have designed the vaccines to protect ourselves at certain level, but still long way to go ourselves to achieve targeted remedies against threats in near future. We salute our COVID warriors and pay homage to all researchers, academicians, scientists whom we lost. Under present scenario, it is very evident to conduct such seminar for sharing of proper knowledge within scientific fraternity about the present day researches leading to promising approaches for better tomorrow. We are fortunate enough to have our guest of honor, Professor Deepak Kumar Kaur, Honorable Vice Chancellor, Sidhu Kanu Birsa University, Purulia, and other eminent speakers who agreed to share their knowledge and valuable time with in the seminar. It is great pleasure that our principal, Shikha Bhavan, Professor Tara Prasad Chattopadhyay, with us. Welcome, sir. I also welcome Principal, Raidighi College and IQSA Coordinator in this session. Once again, I welcome all dignitaries, delegates, faculty members, and participants in this seminar. Now I request our PhD student Devushmita Mukherjee and Kunal Shaha to conduct this session. Thank you very much.
Thank you, ma'am.
Thank you, ma'am.
A very good morning to all the dignitaries, delegates, participants, and guests. I am Kunal Kumar Shaha.
Myself, Devushmita Mukherjee. We are the research scholars of Department of Botany, Visva-Bharati.
We are delighted to be your host, and it's our privilege to welcome you all in this 2 days national seminar on cytogenetics, in vitro culture, and phytochemistry of plants and microbes for sustainable use.
We are very glad to announce that students of Botany Department, Visva-Bharati, will commemorate the inauguration session by a beautiful song of Gurudev Rabindranath Tagore, Kobe Ami Bahir Holam, Tomari Gan Ge. Students,
শোনা যাচ্ছে না কেন?
মিউজিক্যাল ইনস্ট্রুমেন্টের একটু সাউন্ড এসেছিল জাস্ট এক সেকেন্ডের জন্য ওকে
কবে আমি বাহির হলেম তোমারি গান গে, সে তো আজকে নয়, সে আজকে নয়। কবে আমি বাহির হলেম তোমারি গান গে, সে তো আজকে নয়, সে আজকে নয়। ভুলে গেছি কবে থেকে আসছি তোমায় চেয়ে, সে তো আজকে নয়, সে আজকে নয়। কবে আমি বাহির হলেম তোমারি গান গে, সে তো আজকে নয়, সে আজকে নয়। ঝর্ণা যেমন বাহিরে যায়, জানে না সে কাহারে যায়। ঝর্ণা যেমন বাহিরে যায়, জানে না সে কাহারে যায়। তেমনি করে ফেলে জীবন ধারাবে, সে তো আজকে নয়, সে আজকে নয়। তবে আমি বাহির হলে তোমারি গান গে, সে তো আজকে নয়, সে আজকে নয়। কতই নামে ডেকেছি যে, কতই ছবি এঁকেছি যে, কোন আনন্দে চলেছি তার ঠিকানা না পেয়ে, সে তো আজকে নয়, সে আজকে নয়। কত নামে ডেকেছি যে কতই ভেকেছি কোন আনন্দে চলেছি তার ঠিকানা না পেয়ে আজকে নয় সে আজকে নয় খুশবু যেমন আলোর লাগে না জেনে যেমন আলোর লাগে না জেনে রত্নটাই জাগে তেমনি তোমার আশায় হৃদয়
কে?
কবে আমি বাহির বলে
কি হয়েছে?
Thanks, Shreya and Aditi. This is a very beautiful song. This song is from Gurudev's Puja Porjai, connects us to that prime sense of devotion towards a desired destination of life. This lesson is also resonating in our lives.
Now, we would like to welcome the chairman, Professor Swadesh Ranjan Bishwas, the head of the Department of Botany, Visva-Bharati. We are requesting our respected HOD sir to
Declining at rates unprecedented in human history. And the rate of species extinction is accelerating, which will have a serious impact in our own survival. The health of the ecosystem on which we and all other species depend is deteriorating more rapidly than ever. We are eroding the very foundation of our economies, livelihoods, food security, health, and quality of life worldwide. The diversity within species, between species of ecosystem, as well as many fundamental contributions we derive from nature are declining fast, although we are still have the means to ensure a sustainable future for people and the planet. Around 1 million animal and plant species are now threatened with extinctions, many within decades, more than ever before in human history. Deforestation and desertification caused by human activities and climate change pose major challenges to sustainable development and have affected the lives and livelihoods of millions of people. Forests are vitally important for sustaining life on Earth, and therefore we should conserve our forest ecosystem. Traditional medicines continues to play an essential role in healthcare, especially in primary healthcare. Traditional medicines are estimated to be used by 60% of the world's population, and in some countries are extensively incorporated into the public health system. Medicinal plant use is the most common medication tool in traditional medicine and complementary medicine worldwide. Therefore, proper documentation and scientific validation of indigenous knowledge is paramount for growth and development industrially. Today, the world faces a continuous challenge to feed the ever-growing world population as nearly 1 billion people go hungry every day. There is depletion of groundwater because we are tapping more water than we need through pumps for various purposes, which in future the water will become scarce, which will threaten our civilization. The entire world is now gripped by the deadly coronavirus SARS-CoV-2, which has claimed thousands of lives and millions more are affected by it. With no cure or effective vaccine for COVID-19, the world is on the brink of its worst crisis since the last World War. I hope this joint effort will create awareness and provide the right platform for all of us to do innovative research to find solution to various existing problems the world is facing. Lastly, I would like to express my heartfelt thanks to the authority of Visva-Bharati University for approving and providing the facilities for this seminar. As the seminar is online and the transmission relies on the quality of internet, I hope you will bear with us in the event of any interruption in the connectivity. I wish the best to all the participants and the seminar every success. Thank you all.
Thank you, sir, for your kind words. We are honored to have Professor Taraprasad Chattopadhyay, the principal of Shiksha Bhavan Visva-Bharati, as the president of this seminar. And we are requesting him to kindly give his presidential speech in this occasion. Sir, please.
Am I audible?
Yes, sir, you are audible.
Okay. Good morning, everybody. Namaskar. Thank you, Dr. Anjolika Rai, for inviting me to inaugurate the two-day national seminar and to speak a few words on this occasion. Honorable Vice Chancellor Visva-Bharati, Professor Vidyut Chakraborty, Honorable Professor Deepak Kumar Kaur, Vice Chancellor Sidho Kano, Birsa University Purulia, and our guest of honor, respected Dr. Saso Bindu Jana, Principal Raidighi College, and our special guest, respected keynote speakers and plenary speakers, distinguished speakers and chairpersons of different technical sessions, dear Professor Swadesh Ranjan Biswas, Head of the Department of Botany, Visva-Bharati, conveners of the seminar, dear Dr. Anjolika Rai, the joint convener of the two-day national seminar, respected faculty members of Visva-Bharati and Raidighi College who are present online, research scholars, participants, staff members, students, friends, ladies and gentlemen. I am very happy to learn that our botany department, in collaboration with the Botany Department of Raidighi College, is organizing a two-day national seminar entitled Cytogenetics, In Vitro Culture and Phytochemistry of Plants and Microbes for Sustainable Use. Chemical science, I think, has much to do in biological processes. Whatever synthesis or analysis you do in botany may somehow be related with chemical science. Not only chemical science, botany has also relationship with physical processes. Absorption and transmission of light by leaves of any plant is basically a physical process. On behalf of Shiksha Bhavan Visva-Bharati, I welcome you all to this two-day national seminar. Myself, being the principal Shiksha Bhavan, explain the specific nomenclature followed in Visva-Bharati. Here, Bhavana means faculty and principal means dean. Thus, principal Shiksha Bhavana means dean of the Faculty of Science. At the very beginning, I must say that I am a student of physics and my research career stands on microwave and optical communication. So to speak something on cytogenetics is a difficult job for me. The Department of Botany, Visva-Bharati, is one of the leading departments of Shiksha Bhavana, which is vibrant not only in the academic sphere, but also in the socio-cultural area. The Department of Botany has got recognition from the University Grants Commission for DRS SAP Level 2 program. The department has also been selected by the Department of Science and Technology, Government of India for the FIST Level 2 program. The students of Botany department get qualified in NET, SET, GATE, etc. examinations. Many of the students passing out join their research career in many universities and research institutes in India and abroad. Sikha Bhavana has 11 departments and research centers under its control. Visva-Bharati was granted the status of Central University in May 1951 under the Visva-Bharati Act of Parliament. It is an institution of national importance. To know the ideal of Visva-Bharati, we must chant the mantra, Yatra Visam Bhavate Kanidam. In English, where the world makes its home in a single nest. You have already heard, and I am to tell you again, the fact that Visva-Bharati is going to open its second campus in Ramgarh, Uttarakhand. The government of Uttarakhand has donated 45 acres of land to Visva-Bharati, and it has been possible as a result of continuous and sincere effort of when online activities, especially holding seminars, workshops, conferences, came to stand still. Obviously, we had to seek for alternatives. Alternative method was the virtual mode, that is the online mode. To connect so many people all over India and even abroad has only been possible due to the enormous development of wireless communication technology. Had there been no wireless communication, this point-to-multipoint online communication would not be possible. Lastly, I welcome you all again to this two-day national seminar. I would be very happy to see you all in our campus had there been no pandemic. I invite you all to visit Santiniketan with its abundant natural beauty once the pandemic is over. Before closing, I hope this national seminar will be a grand success. Thank you all. Now, over to the next speaker, please.
Thank you, sir, for your valuable words. It's our privilege to have Dr. Shashobindu Jana, the principal of Raidighi College, as a special guest today. He has kindly agreed to be the vice president of this seminar. Now, we would like to request sir to address this seminar. Sir, please.
Thank you. A very good morning to everybody. Today is very special day for Raidighi College.
Carry forward the session.
Our botany department organized a national level seminar collaboration with Visva-Bharati Central University. We are very much grateful to our VC, Dr. Vidyut Chakraborty. He gave us permission for this webinar. Everybody know our college situated in the lineage part of Sundarban. Sundarban is our wisdom of biodiversity. this webinar arranged collaboration with Visva-Bharati. It has been possible by our joint convener Madhumita Majumdar and Dr. Anjalika Rai. At first, I thanks them. A very warm welcome to all of you. Now I welcome to our special guest, Dr. Deepak Kumar Kaur. We are very much grateful for your presence. I give you special thanks and gratitude from our college. He is not only the VC of Birsa Munda University, he is also the General Secretary of our All Bengal Principal Council, Chairman of College Service Commission, West Bengal. He is our guide for our all principals. This seminar organized by our college, the topic cytogenetics in vitro culture and phytochemistry of plants and microbes for sustainable use. I thanks to our chief patron, Dr. Bidyut Chakraborty, Honorable Vice Chancellor, Visva-Bharati. Thanks and respect to our chairman, Professor Swadesh Ranjan Bishwas, Department of Botany, Visva-Bharati. Thanks to our president in this seminar, Professor Tarak Prasad Chattopadhyay, guest of honor, Dr. Deepak Kaur, Senior Professor Narayan Chandra Mondal, Dr. Shishi Chatterjee, Associate Professor, Raidighi College. He is the coordinator of our IQAC. Now I wish all the success for this webinar, and I eagerly waiting for the most beneficial outcome of today's webinar. At last, again, I welcome all of you and wish you all a grand success. Thank you very much. Thank you all again.
Thank you very much, sir. Now, it's time to unveil the very core essence of this seminar, that is the e-abstract book. On this regard, we are glad to request the chairman, Professor Swadesh Ranjan Bishwas, to release the e-abstract book for all of us. Sir, please.
Sir? HOD, sir?
Swadesh da?
Yes, okay, I'm doing that
Okay, sir.
in a moment. Is it visible to you, all of you?
Yes.
Yeah, see the e-abstract book. I'm very happy to release this e-abstract book. This contains abstracts coming from keynote speakers and plenary lectures, and also the poster presentation. I believe that this e-abstract book will be a glorious document of the department and also the university. Please go through this abstract in order to increase your conception and knowledge so that you can proceed with your research works to enhance the fertility or to enhance the essence of the research. Thank you all. Thank you very much.
Thank you, sir. Now, Devoshmita and Kunal, please continue with the session.
Yes, ma'am. Thank you, sir. Hope all of you will be glad to go through the e-abstract book. And now we are glad to have Professor Narayan Chandra Mondol, Senior Professor of the Department of Botany, Visva-Bharati, among us. Sir, please share your valuable thoughts with us.
Thank you, Devasmita and Kunal, and thank you, Anjolika, for sharing this inaugural session with the eminent scientists and botany fraternity and these biological scientists and all the dignitaries. And the president of today's seminar, Professor Taraprasad Chattopadhyay, Dean of Science Faculty or Uddhako Shiksha Bhavan Visva-Bharati. Our guest of honor, Professor Deepak Kumar Kaur, Honorable Vice Chancellor, Sidhu Kanu Birsa University, Purulia. Chairman of this seminar, Professor Swadesh Ranjan Vishwas, Vice President, Dr. Shashabindu Jana, IQC Coordinator of Raidighi College, C.C. Chatterjee, and our very dear Anjolika, Madhumita Majumdar, and all my colleagues here in Visva-Bharati and the faculty members in Raidighi College. So it is my great pleasure to be associated with this 2 days national seminar, which we are organizing through online on the topic cytogenetics in vitro culture and phytochemistry of plants and microbes for sustainable use. Jointly conducted by Department of Botany, Visva-Bharati, and Department of Botany, Raidighi College. So earlier already Swadesh Da, or our chairman, has spoke a lot on different aspects of this seminar and from academic point of view, as well as from realistic point of view, how we are organizing this. And it is really, we are in a challenging phase of time. Otherwise, during this particular January, month of January, this winter month, it would be a festive mode of time. This 2 days national conference in Visva-Bharati, just we cannot imagine this type of, but we have to adapt everything. We have to accommodate everything. So we have to face all the challenges. So at this moment of time, we are facing maximum the challenge posed by this genetic mutation. As per title of this seminar, the genetics, cytogenetics, all these things, and we are the most of this maximum problem we are getting from these changes in the spike proteins of this coronavirus. This RNA virus having its spike proteins on its envelope, these spike proteins have the ability to enter into the this just unlock the ACE receptors of human cell membrane, and it enters into the human cells of this our nasal or respiratory system and cause the damage. And because of this, this thing made a havoc in our society in all over the world and made this pandemic. And maximum impact is occurring on this school boys, college students, and all the academics. So most affected parts are the academic components of faculties, students, and all those things. So we are maximum sufferers, but we should not live. We have to, just being all like all other living organisms, we have to adaptation. So we have adapted to this webinar mode, so with this online mode and virtual mode. So from this challenge, spike proteins and a lot of challenges are there continuously because of our problems, because of our greed, because of unjudicious living style. We are creating a lot of stress to the environment. lot of pollutants to the environment, and because of that, lot of genetic mutations are occurring, and just one single mutation in the gene insulin in the hemoglobin gene, a single mutation, it converts just glutamine, it converts just a single mutation made a sixth amino acid. It is changed from valine to valine is forming, and it becomes sickle cell anemia. So sickle shape. So hemoglobin, the RBC molecules, its normal shape is changed. It becomes sickle, and it cannot hold the hemoglobin in the RBC, and so a single genetic mutation is there. A lot of such examples are there. This Philadelphia chromosome. So 9th chromosome and 22nd chromosome, there is a exchange of this contents, and because of all sorts of stress, we are generating the teratogenic compound, this carcinogenic, we are creating by our activities. So these things are creating a lot, creating havoc, and causes these mutations, and cytogenetics comes in this way. So we have to work with the help of this molecular biology, the innovative techniques. The scientists are working a lot on these, and then we are losing a good amount of biodiversity from this earth. And here comes this in vitro repropagation, all these conservation methods, cytogenetics, these tissue culture techniques, and these things are very important. So in today's context, in global context, in our lifestyle context, whether we are doing wrong or right, we do not know. But actually, these things, in reality, these things happen. We are creating a lot of problems to this environment, and because of this, we are losing biodiversity. And at the same time, the scientists with some people, they gather together, they just come in a dais in different forum, and they are thinking, the scientists, they are sweating a lot in the laboratory, they are sacrificing their sleeps in the night, and they are doing, they are not participating in the social festivals and social programs, and they are doing a lot and doing this tissue culture technique and these molecular, these novel discoveries and all those things. So because of some good thoughts, some good sacrifices, and these things, we can escape from such problems. So, and these are the problems I am telling. And we can, at the same time, this biotechnology, we can use these genes. So once a modification is there, so Gandhiji, it was published in a newspaper, the Gandhiji, he will remain immortal in our minds. Somehow T was vanished, deletion was there, so Gandhiji will remain immoral. Just think what happened, a single mutation. So similarly, so genetic, just a single change, so we can, with the change, a lot of problems are there. At the same time, we can put some good genes in good places, so we can get a lot of good things. So I do not criticize this allopathy medicine. Allopathy medicines at this moment of time, or if we use judiciously, proper use, it can, at this moment of time, there is no other alternative, at least for major people, for most of the people, allopathy medicine is the final rescue. But this allopathy medicine has a lot of problems, lot of side effects. Amphotericin B, it has a lot of renal failure, a lot of problems. But just to control mucormycosis, this black fungi, which is an outcome of this corona, this mucormycosis, we can, so azole drugs, which are fungistatic in nature, cannot control this fungus. But amphotericin B, which is a whose mode of action is lysis, which can lyse the cell, destroy the cell membrane, is the final rescue for this black fungi. And it has a lot of renal toxicity. So like this amphotericin B, lot of this allopathy medicines or modern medicines, they have side effects. And again, we like yesterday I had heard a lecture from our ICCR chairman, who is representing all the eastern parts of the Asia and all different countries, and he just the soft things, this behavior and all other things, good things like yoga, like Ayurveda, all things which can contribute to the world from India. So again, the potential of plants, we should, if we can use the potential of plants in a judicious manner, not only plants, the potentials of microbes, a lot of good bacteria are there, which are known as probiotic bacteria. So a lot of good bacteria are there. They are metabolites, the metabolites of plants, which can be used in a judicious manner, and their propagation, their multiplication, incorporation of gene, proper gene. So these things will help us a lot. So a lot of plant metabolites can be derived, lot of microbial metabolites can be derived, which can be used for medicinal purposes, like artemisinin. So the malaria drug, to control malaria, earlier we are dependent on this quinine. Recently, a Chinese group, they have discovered UU2. So his group discovered this Artemisia from Artemisia plant. They have discovered another metabolite which can control this malaria. So there is huge prospect, and we can, if all continuously we can improve the genes. Like when earlier Alexander Fleming discovered penicillin, it could produce only 40 units of penicillin in a certain volume of thing. Now this same the species is changed. Penicillium chrysogenum, earlier it was Penicillium notatum. So now with the help of genetic improvement, these different types of mutations, they just protoplast fusion, different types of mutation in the genes, and we can achieve up to 3,000 units instead of 40 units during Alexander Fleming. Today we can, in our industry, they produce 33,000 units of penicillin using some modified systems, in modified this technology, modified this, mainly the genetic content of the penicillium has been changed by the scientists. So we have to think in a proper way so that we can identify the negative effects of genes which are created by our callousness or by our illogical or by our irrational or by our greedy activity, and we are generating a lot of pollutants, a lot of stresses, and these lot of stresses are creating a number of mutations. Sometimes all these mutations are not created by our byproducts. These are created naturally. Anyway, so we have to identify these mutation negative things, and we have to find out the ways to overcome these things, and we have to find out how we can do good things using these genetic aspects, and using another green methods. These plants are considered as green methods, not only in the sense of they are green in color, because they do not create pollution, they do not create harm to the environment. That's why I'm telling this green method. So in contrast to allopathic medicine, so Ayurvedic medicine, so the plant metabolites, there is no, I do not know why we should not use any terminology for Ayurvedic or this allopathic, or thus these plant metabolites are used to this Artemisia. Habib can correct me, species. I'm forgetting, I forgot the species.
Artemisia annua.
Artemisia annua. So this Artemisia, its metabolite is used in allopathy also. So it's not a problem. So we have to do all the biology in a judicious manner. And I am very happy to share you that our colleague, the chairman of today's seminar, Professor Swadeshwar Ranjan Biswas, has worked a lactic acid bacteria, probiotic bacteria metabolite, which can combat, which can interact, which can inhibit the spike protein functions of this coronavirus. So these are the important steps which we can do for the society and for the humankind. So we have to find the different ways. How can we use the potential? How can we exploit the benefits of different types of microorganisms, plants, all those things? So in this forum, I hope with the deliberation of so many eminent scientists, Professor Deepak Kumar Kaur, our very beloved Deepak Da. So he is the Honorable Vice Chancellor of Sidhukan University. He has done a good amount of work on cytogenetics, and he will he will put some light on this aspect in a better way. So, and our Nirmal is there. He has done a good amount of work on tissue culture. So all the different scientists are here. So, and all the students are here. So we can share our knowledge. We can share our knowledge with the botanic fraternity of different scientists, different groups. Immediate earlier, Professor Taraprasad Chattopadhyay has that the wireless. This is the importance of wireless. If there is no wireless, so we cannot gather today in this seminar. So the importance of all the science, we have to realize the importance of all the science. And I hope, I believe, and I fully trust that this communication, the exchange of knowledge organized by these two important institutions will definitely give an fruitful outcome. And with this hope, I would like to conclude my speech here. So again, I welcome all the scientists, all the dignitaries, all the delegates, all the students, and my heartfelt thanks to all of them, and love you all. Thank you.
Thank you, sir, for this informative input. And we are immensely elated to have Professor Deepak Kumar Kaur, the Honorable Vice Chancellor, Sidhu Kanu Birsa University, Purulia, as our guest of honor, and we are requesting sir to enrich this seminar with your kind words. Sir, please.
শোনা যাচ্ছে
হ্যাঁ স্যার
Yes, sir.
okay, then I had to give 2 times speech, right? The keynote address will be in the next session.
Yes, sir.
Okay, thank you very much. Good morning everybody in this virtual platform. Pranam to Gurudev Rabindranath Tagore, the founder of Visva-Bharati. First of all, I wish to congratulate the Department of Botany of Visva-Bharati, all the faculty members, scholars, students in organizing this 2 days national seminar in association with the Department of Botany of Raidighi College in vitro culture and phytochemistry of plants and microbes for sustainable use. At the same time, I wish to express my sincere gratitude to the organizing committee for inviting me on this occasion. সম্মানীয় অধ্যক্ষ শিক্ষা ভবন বিশ্বভারতী প্রফেসর তারাপ্রসাদ চট্টোপাধ্যায়, respected head of the Department of Botany of Visva-Bharati, Professor Swadesh Ranjan Biswas, respected principal of Raidighi College, my friend Dr. Shashabindu Jana, respected Professor of Botany Narayan Chandra Mondal of Visva-Bharati, respected IQAC coordinator of Raidighi College, Dr. C.C. Chatterjee, respected senior retired professor, Professor Nirmala Banerjee, a good friend of mine, distinguished speakers of plenary lecture sessions, respected participants, research scholars, eminent scientists, distinguished dignitaries, dear students, and of course the respected conveners, Dr. Anjalika Rai and Dr. Madhumita Majumdar. I express my heartiest congratulations and thanks to the entire fraternity who are being related with this seminar. As I begin to speak later on in the keynote keynote lecture session. So wish this 2 days seminar a grand success. Thank you very much.
Thank you very
Thank
much.
you very much, sir, for enriching us. We are delighted to announce that we have Dr. Urnia Bishwas, IQAC Joint Coordinator, Raidighi College with us. Now we are requesting ma'am to address this session. Over to you, ma'am.
Thank you. Good morning to all respected dignitaries, honorable guests, and dear students. I'd first like to congratulate the organizers, especially the joint conveners, Angelika ma'am and Madhumita ma'am, for this great collaborative initiative. It's indeed a great collaboration between two departments, one from a central university and another from our rural college. And these are probably one of the few silver linings of this depressing pandemic, that science has not stopped, science is progressing, scientific discussions are on, and such platforms, such interactive platforms are really great to exchange ideas, and it's also a great opportunity for our students. Because, you know, among the developing countries that are racing to achieve scientific excellence in this age, India is a leading contestant, no doubt. But earlier, physical science, maths, engineering, medicine, these were considered to be higher scientific endeavors than studying general biology. Academically strong students were usually directed to careers in medicine and engineering. And even in recent times, India's economic boom has mostly been driven by its IT and service industries. However, the policymakers of the nation have now started appreciating the important role, the very crucial role that modern biology is playing in this decade and is predicted to play in the next few decades. So India is heavily relying on biological research for its scientific future, and public interest in biology has also greatly increased, especially in this pandemic. But unfortunately, India's footprint in modern life sciences is still restricted mostly to few urban institutes and universities. You know, our huge rural population, they are still ignorant and mostly clueless about the giant leaps biology has taken. We have vast natural resources, for example, where our college is in the Sundarban region, and it's blazed with such diverse flora, fauna, microbial life, etc. But they mostly continue to run to west. We don't utilize our natural products that much for their potential applications. And people are unaware in the backward village regions where that how modern biology can solve their problem of deadly infections or genetic conditions, food insecurity, pollution, climate change. I mean, biology probably has the answer to all. So this kind of scientific interaction between these two departments today, I mean, they are going to play a great role in making biology more popular, you know, both in the urban and the rural population. And that's the only way to make biology an all-inclusive enterprise in global perspective for our country. So it's, once again, it's a great initiative, and I'm sure it's going to be a very enriching journey of 2 days for all of us. So I wish it a grand success. Thank you.
Thank you, ma'am, for your kind words. We have reached the very end of this inaugural session, and we would like to request Joint Convener of the seminar, Dr. Madhumita Majumdar, Head of the Department of Botany, Raidighi College, to convey the vote of thanks. Ma'am, please.
Thank you. Very good morning, everybody. Honorable guest of honor, respected President, Vice President, Chairman, distinguished speakers, respected members of Advisory Committee and Organizing Committee of today's National Seminar online on 2022 on Cytogenetics in Vitro Culture and phytochemistry of plants and microbes for sustainable use. It's my privilege to have been asked to propose the vote of thanks on this inaugural session. On behalf of the organizing committee, I, Dr. Madhumita Mojumdar, extend my heartfelt thanks to the honorable guest of honor, Professor Dr. Deepak Kumar Kaur, Vice Chancellor, Sidhu Kanu University, Purulia, for gracing this us. We are really one of our keynote speaker today. Thank you so much, sir. I express my deep sense of gratitude to our respected President, Professor Tara Prasad Chattopadhyay, Principal, Shikha Bhavan, Visva-Bharati, and special guest, Dr. Shashubindu Jana, Principal, Raidighi College, for their constant support and encouragement. I am also thankful to the authorities for allowing us to conduct this program. I am thankful to Professor Swadesh Ranjan Biswas, Head, Department of Botany, Visva-Bharati, for having the vision to organize this today's national seminar, where we have enthusiastic participants from all across the country. I sincerely thank Professor Narayan Chandra Mondol, Senior Professor, Department of Botany, Visva-Bharati, for his encouraging speech. Special thanks to Dr. Arunima Biswas, IQAC Diet Coordinator, Raidighi College, for his constant support and words of encouragement. We are highly grateful to all the distinguished keynote and plenary speakers for gracing this today's national seminar with their presence. I am extremely thankful to all the faculty members of Department of Botany, Visva-Bharati, and Department of Botany, Raidighi College, without whom it would not have been possible to organize such type of seminar. Last but not the least, we appreciate such active participation by the participants. With that, we come to the end of this inaugural session. Now we will hear the closing song performed by the student of Botany, Visva-Bharati. Thank you.
Thank you, ma'am. Now I would ask our departmental junior students to deliver the closing song, Jagoto Jure Udaro Shure, Anandogan Baje, to mark the end of the inaugural session. Following this, we will proceed towards the keynote and plenary lecture sessions. So all are requested to stay in the same link. Thank you.
জগত জুড়ে উদার সুরে আনন্দ গান বাজে জগত জুড়ে উদার সুরে আনন্দ গান বাজে সে গান কবে রং বাজিবে হিয়া মাঝে জগত জুড়ে উদার সুরে আনন্দ গান বাজে বাতাস জল আকাশ আলো সবারে কবে বাসিব ভালো বাতাস জল আকাশ আলো সবারে কবে বাসিব ভালো হৃদয় সভা জুড়িয়া তারা বসিবে নানা সাজে হৃদয় সভা জুড়িয়া তারা বসিবে নানা সাজে জগত জুড়ে উদার সুরে আনন্দ গান বাজে নয়ন দুটি মেলিলে কবে পরানো হবে খুশি যে পথ দিয়া চলিয়া যাও সবারে যাব তুষি নয়ন দুটি মেলিলে কবে পরানো হবে খুশি যে যে পথ দিয়া চলিয়া যা সবারে যাব তুষে রয়েছ তুমি এ কথা কবে জীবন মাঝে সহজ হবে রয়েছ তুমি এ কথা কবে জীবন মাঝে সহজ হবে আপনি কবে তোমারি নাম নিবে সব কাজে আপনি কবে তোমারি নাম নিবে সব কাজে জগৎ জোরে উদার সুরে আনন্দ গান বাজে জগৎ জোরে উদার সুরে আনন্দ গান বাজে
Thank you, Shayan Aditi. Finally, we have arrived in our most awaited keynote lecture session. I would like to request Professor Swadesh Ranjan Bishwas, Head of the Department of Botany, Visva-Bharati, to kindly chair this session. Sir, please.
Sir,
Hello, sir.
Sir is joining with us. Okay, few minutes.
okay, ma'am.
Hello, Dr. Anjulika, am I audible?
Yes, sir.
Sir, you are audible.
You're audible, sir.
Good morning. We are going to start the next session. The keynote speaker, the renowned scientist, Professor Deepak Kumar Kar, Vice Chancellor, Sidhu Kanu Birsa University, Purulia. Let me say a few words for introduction. Professor Kar obtained his BSc 1977, MSc 1979, and he was awarded PhD in 1985. He has a 30 years of research experience. And research experience, doctoral and postdoctoral work together 10 years. He has a lot of administrative experience. He's currently serving as Vice Chancellor Sudhakano Birsa University. And chairperson, he is also the chairperson West Bengal College since 2014. And University in the time 2019 college during the year 2009 to 2014. He has been selected as the chairman of Ashutosh College Training Center in 2009 to 2014. And he also served as HOD Botany and Microbiology Ashutosh College in 1998 to 2009. He has published a quite number of research papers in international journals of repute. 14 research papers in reputed journal, international, 5 international 5, and national publication 9. And he has published 10 abstracts in Proceedings of Symposium, International 3 and National 7. And he has published 10 books. And regarding seminar and conferences and workshops, he attended 55 and organized 20 seminar conferences. He has been an active member of Syndicate and Executive Council, Court of Universities of West Bengal. He has been active as member of various scientific societies, academic bodies, professional organization, General Secretary. So I welcome you, sir. Professor Deepak Kumar Kar. This is going to be our keynote address to be delivered by our respected Professor Deepak Kumar Kar. Sir, please.
Thank you. Thank you, Professor Biswas. Respected chairperson, আমাকে শোনা যাচ্ছে তো?
Yes.
Okay, okay.
You are audible.
Okay. Respected chairperson of this keynote session, Professor Swadesh Ranjan Biswas, distinguished scientist, head of the Department of Botany, Visva-Bharati. Thank you, sir, for your kind words. May I start the presentation, sir? Okay, already. Just a minute. I need to minimize it. Dr. Madhumita or Dr. Anjalika?
Yes, sir.
Are you sharing it?
Yes, sir. It is visible.
Already? Okay. It is visible to all? Is it visible to all?
Yes, sir.
হ্যাঁ স্যার দেখা যাচ্ছে।
Yes,
দেখা
we can
যাচ্ছে?
see.
দেখা যাচ্ছে, হ্যাঁ।
আচ্ছা, স্ক্রিনটা বড় করে দেবেন?
বড়ই তো আছে।
বড়ই আছে, বড় আছে এটা।
হ্যাঁ।
না, ফুল স্ক্রিন করে দেওয়া যাবে না?
হ্যাঁ স্যার করে দিয়েছি।
স্যার ফুল স্ক্রিনই
আর
আছে।
তো ল্যাপটপে তো ফুল স্ক্রিন দেখা যাচ্ছে আর কি।
ফুল স্ক্রিন দেখা যাচ্ছে। Okay.
হ্যাঁ।
Thank you again for inviting me and giving the scope to deliver the keynote address on cytogenetics, in vitro culture, and phytochemistry of plants and microbes for sustainable use. As the keynote address demands a particular at a particular depth or particular dimension, but the topics or the fields are so diverse. I don't know how will I do the justice to the audience. Thank you. Next slide, please. Next, please. Dr. Madhumita. Hello.
হ্যাঁ। Yes, sir.
And next slide, please.
হ্যাঁ স্যার, নেক্সট স্লাইড দিয়েছি।
দিয়েছেন? আচ্ছা, আমার এখানে কেন আসছে না? দাঁড়ান, এক মিনিট দেখে দাঁড়ান। আমার এখানে কেন আসছে না বলুন তো?
মধুমিতা আসছে না কিন্তু।
আসেনি কিন্তু
Just
আমি শেয়ার করব
আপনি শেয়ার করতে পারলে ভালো, কেননা
হ্যাঁ,
এখানে এই অঞ্জলিকা আমার ল্যাপটপও কিন্তু ওটা মুভ করেনি, অরিজিনাল স্লাইডটাই আছে।
হ্যাঁ
আচ্ছা
আমি
স্যার,
কি শেয়ার করব
হ্যাঁ হ্যাঁ স্যার, তাহলে আমি দেখছি, আপনি তাহলে স্যার শেয়ার করুন স্যার, আমি
আপনি
আমি
যদি পারেন
চেষ্টা
তাহলে
করছি
ভালো
আমি
হয়।
চেষ্টা
হ্যাঁ হ্যাঁ
আমরা ওয়েট করছি,
আমি-
আপনি শেয়ার করুন।
আমি চেষ্টা করছি আমি চেষ্টা করছি দাঁড়াও।
নেটওয়ার্কের সমস্যা এখান থেকে।
দেখা যাচ্ছে কি দেখা যাচ্ছে
হ্যাঁ স্যার দেখা
হ্যাঁ
যাচ্ছে।
হ্যাঁ স্যার, দেখা যাচ্ছে।
পরিষ্কার দেখা যাচ্ছে।
আমি এখানে একটা নেট ইউজ করছি, স্যার ওখানে একটা নেট ইউজ করতে আরো বেশি অসুবিধা হবে।
না না কিছু গন্ডগোল হচ্ছে My God, কিছু একটা অসুবিধা হচ্ছে।
স্যার দেখা যাচ্ছে কিন্তু পুরো।
না আমি তো মুভ করতে পারছি না, পরের স্ক্রিনে পরের স্লাইডে যেতে পারছি না তো। আমি বেরিয়ে যাই নাকি?
না, আপনি ওই অ্যারোটা দিন না, রাইট সাইডে যে অ্যারোটা, সেটা টিপলেই তো যাবে।
হ্যাঁ সেটা তো টিপছি, যাচ্ছে না তো।
তাহলে গন্ডগোল হচ্ছে কোথায়।
দেখুন তো আপনাদের স্ক্রিনে।
হ্যাঁ হ্যাঁ হ্যাঁ, এবার চলে এসছে, এবার চলে
আচ্ছা
এসছে।
আমি তাহলে পরে ওই মানে ওইটা দিয়ে যাচ্ছি, মানে প্রত্যেকটা করে করে যাচ্ছি আজ। এনিওয়ে
হ্যাঁ হ্যাঁ
দেখা যাচ্ছে, সবার দেখা
হ্যাঁ
যাচ্ছে,
হ্যাঁ, পরিষ্কার দেখা যাচ্ছে।
সবাই পাচ্ছেন দেখতে তো?
হ্যাঁ স্যার,
হ্যাঁ স্যার,
পাওয়া
দেখা যাচ্ছে
যাচ্ছে।
স্যার।
দেখা।
স্যার সেরকম হলে
Full
একটু
screen-
ভিডিওটা অফ করে, হ্যাঁ স্যার, হয়তো নেট।
না আমি ফুল
না না, অসুবিধা হচ্ছে না, ঠিক আছে।
আমি
হ্যাঁ, হ্যাঁ।
ফুল স্ক্রিনটা চাইছিলাম, ফুল
মুভ
স্ক্রিনটা আসছে
করছে
না।
না।
এনিওয়ে আচ্ছা, the national seminar is the three very significant areas of plant sciences, namely cytogenetics, in vitro culture of plants, and phytochemistry of plants and microbes for sustainable use. Cytogenetics, both classical and molecular, in vitro tissue culture techniques, and characterization, extraction, and use of phytochemicals have made immense contribution in academics and of course economical to us. আমি নেক্সট স্লাইডে গেলাম, দেখা যাচ্ছে পাচ্ছেন?
হ্যাঁ 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 হ্যাঁ
সাইটোজেনেটিক্স পাস্ট টু প্রেজেন্ট দেখা যাচ্ছে?
হ্যাঁ, পরিষ্কার দেখা যাচ্ছে।
Okay, okay, okay. The field of plant cytogenetics was heavily influenced by Barbara McClintock's pioneering work on maize. Her method of unequivocal identification of individual chromosomes permitted major discoveries regarding the structure and dynamic behavior of the maize genome. Using carmine-based chromatin staining procedure, she showed that all of the individual chromosomes could be uniquely identified from a single meiotic nucleus with a combination of the matrices, the relative lengths, and the arm ratio of chromosomes. Here is the paper in Science in 1929 of Barbara McClintock, Chromosome Morphology in Geomates. Plant species with similar sized chromosomes require the development of additional techniques for unmistakable cytogenetic resolution and the karyotyping. Development of chromosome bending techniques greatly improved the usefulness of somatic chromosomes, which are significantly easier to acquire than the meiotic chromosomes. Kasparson in 1968 used the fluorescent dye quinacrine to produce additional bending patterns, commonly called Q bands, on plant chromosomes. Here is the bending, quinacrine bending. The first one is Giemsa bending. Next one is quinacrine bending. C, the reverse bending, commonly called R bending, and the last one is the C bending, the centromeric bending. আমাকে একটু অন্য মেথডে প্রেজেন্টেশন করতে হচ্ছে, as the arrow is not helping to proceed. Okay, no problem in it. আপনারা দেখতে পেলেই হলো। এটা পাচ্ছেন তো, মলিকুলার সাইটোজেনেটিক্স স্লাইডটা দেখতে পাচ্ছেন তো? হ্যালো হ্যালো,
হ্যাঁ দেখা যাচ্ছে দাদা।
ওকে ওকে ওকে, আচ্ছা। To address the limitations associated with classical cytogenetics and to overcome those, the molecular cytogenetics started exploring the development of in situ hybridization, comparative genomic hybridization techniques opened opportunities for cytogenetic analysis of any species regardless of its inherent chromosome morphology. Genome mapping, two types, physical maps and genetical maps. The physical mapping uses the molecular biology techniques to examine DNA molecules directly in order to construct maps showing the position of sequence features, including genes. The most important techniques being the restriction mapping, is in situ hybridization, fluorescent in situ hybridization, sequence stacked site mapping also. The development of in situ hybridization is technique developed by Pardo and Gall in 1964 opened up opportunities for cytogenetic analysis of any species regardless of its inherent chromosome morphology. In plants, the use of radioactive tracer or the modified nucleotides to make yeast probes permitted microscopic visualization and localization of complementary sequences in cells and nuclei and on individual chromosome. Here is the yeast plate, localization of repetitive DNA sequences following yeast. The first one is the metaphase plate and interphase nuclei of field bean. B is the localization of repeats on field bean chromosomes. Both C and D, the repeats on field bean chromosomes. E, the slide E, the localization of telomeric repeats on field bean chromosomes. The slide F, the group D wheat chromosomes after localization of repeats. And the slide H, the last one, which is the polymorphism in banding in wheat chromosome 3b. P is the fluorescence in situ hybridization, which uses fluorescent probes that bind only to those parts of the chromosomes which have a high degree of sequence similarity. It aids in gene mapping, toxicological studies, analysis of chromosome structural aberrations, as well as the ploidy determination. Although PIS is commonly used to map unique or low copy number sequences, it is also used to localize the repetitive sequences to produce chromosome recognition cocktails or explore genome relations in polyploid as well as in closely related plant species. The broad application of PIS in structural, comparative, and functional genomics place plant cytogenetics in a unique position to complement, to accelerate, or to guide the plant genome research. The piece on superstress chromosomes, the high resolution piece on superstress flow sorted plant chromosomes, the technique allows longitudinal extension of chromosomes of more than 100 times their original metaphase size, is especially attractive for plant species with large chromosomes, whose pachytiny chromosomes are generally too long and heterochromatin appears too complex for PIS analysis. This is the high resolution piece on wheat chromosome 3b with a probe for wheat bacterial artificial chromosome. The chromosomes 3b, the top one, the chromosome 3b was spontaneously stretched to 1.23 fold after shorting and air drying. And the bottom one showing the distance between inner borders of these signals increased to 41.7 micron, including indicating 208.5 fold improvement in spatial resolution. Sorry. Fiber piece which greatly improves the sensitivity and resolution of the fish technique and allowed the mapping of probes that are 1 kb to 1 MB apart. This is the fiber piece procedure being adopted. This is the piece mapping on chromosomes and extended DNA fibers in cotton, showing the telomere, the red signals on metaphase chromosomes of Gossypium hirsutum, Herbaceum, and Arboreum. Piece as a tool for chromosome identification. Piece signals derived from 2 repetitive DNA probes that allowed identification of all the 21 chromosomes in hexaploid plant, as done by Pedersen and Lange in 1997. Here is the image of the genome A, B, and D. Another application piece for mapping the eukaryotic genomes, the physical mapping of the 5S rDNA and the 45S rDNA in Chrysanthemum and other related genera, as done by Hussein. Going to the image. Yes, the piece signals of 5S and 45S rDNA.
অঞ্জলিকা তুমি মিউট করে রাখো।
আমরা এখান থেকে শেয়ার করছিলাম, তারপর আমরা দিতে পারছি না, উনি যখন
অঞ্জলিকা
মানে ফুল স্ক্রিনে আসছে না।
অঞ্জলিকা তুমি মিউট করো, তোমার এটা মাইক্রোফোনটা স্বদেশেরটাও মিউট করো।
Professor Kor, please make it a full screen. Use the option and click to make it a full screen, please, sir.
That I could not, because I am not getting the option of the full screen here.
Okay, sir.
Okay, piece signal. This is the piece signals of both 5S and 45S rDNA loci on metaphase chromosomes, simultaneously probed. The 5S ribosomal DNA has been labeled with biotin, and 45S ribosomal DNA labeled with dioxygenine. The first one is in Chrysanthemum, second one is Tenaciatum, C and D with the Artemisia, Argenthemum, and the last one is Nipponanthemum. Yes, this is the piece-based karyotyping and the phylogenetic analysis. Piece-based chromosome identification systems can be used for karyotyping. Several repetitive DNA probes generate specific hybridization patterns on chromosomes of wheat and its related species. Comparison of the fish karyotypes of Triticum and Agilops species provided us the chromosomal evidence of the evolutionary relationship between these species, as shown by Badeva et al. And another application is the mapping and isolation of gall-means resistance gene in rice by the PIS technique. With the use of PIS, rice genes have been mapped and isolated, and that contribute towards the gall-means resistance. GM resistance, gall-means resistance survey of a population as done by Zhou Wang. This one is the GIS, the genomic in situ hybridization, a special type of piece that uses genomic DNA of a donor species as a probe in combination with an excess amount of unlabeled blocking DNA, provides a powerful technique to monitor chromatin integration during interspecific hybridization. In addition, the GIS technique allows the study of genome affinity between polyploid species and their progenitors. This is a study of interspecific hybrids of Musa. The analysis of meiotic behavior was performed by means of GIS in interspecific hybrids of cultivated species of banana by Gerardi et al. I don't know whether, yes, না এটা তো হয়ে দেখালাম না, এখানে যাইনি না, just a minute.
আপনি আর ওটাকে দিয়ে নিচের দিকে নামুন, আপনি উপর দিকে উঠে যাচ্ছেন, নিচে যান, আরো নিচে যান, আরো নিচে যান, তারপরের পরেরটা পাশের ক্লাস্টারটা নিচের দিকে নামান।
দাঁড়ান দেখছি, এটা খুব মুশকিল হয়ে গেল। এনিওয়ে করে নিচ্ছি। কত দূর গেছিলাম? এখানে এসে গেছিলাম না, এর তো যাইনি, এখানে ছিলাম না, দেখুন তো, হ্যাঁ এখানেই ছিলাম, তাই তো, রাইট। The study of interspecific hybrids of Musa. This is the image. GIS on meiotic metaphase 1 of interspecific triploid Musa cultivar. The plate B is with B genomic DNA revealed in red, and plate C, a genomic DNA revealed in green, and D and F after superimposition of the three colors. Kindly note the arrows which indicate the homologous bivalence, and the arrowheads 3 in the plate F that indicate homologous multivalence, trivalent. This one, trivalent. Yes, this is the next one, the array comparative genomic hybridization. Comparative genomic hybridization is a molecular cytogenetic method for analyzing the CNVs, copy number variations, relative to ploidy level in the DNA of a test sample compared to the reference sample without the need for culturing the cells. The aim of this technique is to quickly and efficiently compare the two genomic DNA samples arising from two different sources, which are most often closely related, because it is suspected that terms of either gains or losses of either whole chromosomes or subchromosomal regions. And through the use of DNA microarrays in conjunction with CGS technique, comparative genomic hybridization technique, the more specific form of array CGAs has been developed, allowing for a locus by locus measure of copy number variations with increased resolution as low as 100 kilobases. Here is the characterization, the paper on characterization of hemizygous deletions in citrus using array comparative genomic hybridization. Use of array comparative genomic hybridization for the successful identification of genes included in a hemizygous deletion induced by fast neutron irradiation on citrus, and the local gene collinearity in the species were higher with populus trichocarpa than with the phylogenetically closer Arabidopsis thaliana. And this is the genome mapping by genetic maps. Genetic mapping is based on the use of genetic techniques to construct maps showing the positions of genes and other sequence features on a genome. Genetic techniques include crossbreeding experiments, or in the case of humans, the examination of pedigrees, the family histories, and the DNA markers are used for genetic mapping, which are, these are the different molecular, this is about the molecular markers, the DNA markers, which are the segment of DNA that can be detected through specific laboratory techniques. For detection of markers, either restriction enzymes or PCR or their combination are used to generate, to amplify the DNA sequences that are linked to a heritable trait, such as the yield or the disease resistance. Mass the marker assisted selection allows heritable traits to be linked to the DNA segments that are responsible for controlling that trait. These segments of DNA or QTL, quantitative trait loci, can be detected through specific laboratory techniques. Here is the different types of molecular markers: RABLB, RABD, SSR, STS, SNP. Their comparison in term of whether they are PCR based or not, the polymorphism abundances, dominance, reproducibility, and the automation and the running cost of the markers. These are the examples of some genes in different cereals, their traits, and the genes from parent 1 and the genes from parent 2, and the section stage and the marker involved. In barley, rice, wheat, there are a few examples of the concentrates of the crops and their parents. DNA microarray. DNA microarray allows the rapid and simultaneous screening of many thousands of genes. DNA segments from genes of known sequence, a few dozen to hundreds of base pairs long, are synthesized directly on a solid surface by a process called photolithography. Thousands of independent sequences are generated, each occupying a tiny part or a spot of a surface measuring just a few square centimeters. The pattern of sequences is pre-designed with each of many thousands of spots containing sequences derived from a particular gene. And once constructed, the microarray can be probed with messenger RNA or complementary DNA from a particular cell type or cell culture to identify the genes being expressed in those cells. Here the micro array based single feature polymorphism SFP study. There is a list of SFP studies conducted in various crops, wheat, rice, and barley. The resources used and the features. Diversity array technology, which is a high throughput microarray hybridization based technique that allows simultaneous typing of several hundred polymorphic loci spread over a genome without any previous sequence information about these loci. And this diversity array technology markers have been developed in rice, in barley, and now on large scale wheat, and were extensively utilized for the study of genetic diversity, preparation of integrated framework linkage maps, and association mapping. Diversity array technology markers have also been used for QTL mapping, for fusarium head blight in wheat, and for leaf pubescence in barley. Here is a list of diversity array DRT studies conducted in different crop plants. Their markers, shell features. Yes, this is the red, the restriction associated diversity markers. A variety of microarrays, complementary DNA or oligonucleotide arrays have also been used to develop the red markers for study of genome-wide variations associated with restriction sites for individual restriction enzymes. And for this purpose, first a genome-wide library of red tags is developed from genomic DNA, which is then used for hybridization onto the chosen microarray to detect all restriction site associated variations in a single assay. Red markers has been successfully developed in a number of organisms, including fruit fly, zebrafish, neurospora, and is being used in laboratories working on higher plants. This is the databases for detecting various chromosomal abnormalities. It is a historical timeline in the development of databases which have been produced for researchers to deposit, to share, and to compare the inconsistent information over the past 20-25 years. And this is the softwares used in cytogenetics over the last 30 years. There are timeline in the development of softwares, which is commonly used to detect a variety of chromosomal aberrations over the last 30 years. This is the table of some major crops which have been improved through cytogenetic approach. The crop type, the trade acquired in rice, wheat, sickle cereal, sorghum, gossypium, in pea, banana, capsicum, sugarcane. Now, in most of the cases, enhanced agronomic and yield performances has been achieved, along with resistance, number of grain protein, superior increased tiller numbers and superior grain protein, gluten contents. This is the last one in the cytogenetic part, the post-genomic perspective on molecular cytogenetics.
He said he can't see me. He's asking me to put on my video. Here it is. It's their problem, no? Yes.
from transcriptomics to proteomics and to metabolomics. Much more data has been collected, but so far there has been no fundamental change at the conceptual level, as the relationship among genomics and other omics simply reflects the relationship of information layers between genotype and phenotype, and it has become much more easier to obtain data, but incredibly difficult to interpret them. Anyway, I am moving to the next section of my address, that is the in vitro culture. Just a minute, please. Okay. In vitro culture, the tissue culture, is the in vitro aseptic culture of cells, tissues, organs, or of whole plant under controlled nutritional and environmental conditions, often to produce the clones of plants. The controlled conditions which provide the culture and environment conducive for their growth and multiplication, and these includes the proper nutrients, the pH, the medium, adequate temperature, aeration, and the liquid environment. Plant tissue culture technology is being widely used today for large-scale plant propagation. Here is the schematic presentation of major areas of plant and tissue culture and some field of application. You see the plant cells is with the capacity potentiality of totipotency, the inherent capacity of the living cells. We can exploit that and through the different device techniques like meristem culture. The plants can be regenerated, developed through meristem culture from the explant, or through the formation of callus by organogenesis or by embryogenesis. We can regenerate the new plants, as well as we can macerate the plant tissue into cell suspension, and the cells in suspended condition may follow the embryogenetic pathway. As we all know, the somatic embryogenesis is very much common in plant system. So plants can be regenerated through somatic embryogenesis. And nowadays in crop plants, the production of artificial seed is a common practice. And the cell suspension can also be used for the production of the secondary metabolites through fermenter culture. And another technique in plant tissue culture is the protoplast isolation culture and subsequent hybridization. The protoplast may be directly subjected to embryogenesis to generate the plant, or may be used in fermenter for production of secondary metabolites, or maybe hybridized with the closely or unrelated species to develop the somatic hybrids, the important technology in plant breeding today. And another technique in plant tissue culture is the anther and microspore pollen culture through androgenesis. The haploid plants can be produced either through callus formation or directly by somatic embryogenesis. The in vitro culture, regarding its application, apart from their use as a tool of research, plant tissue culture techniques have become a major industrial importance in the area of plant propagation, disease elimination, plant improvement, as well as in the production of secondary metabolites. Apart from that, for germplasm conservation, the endangered, threatened, and rare species have successfully been grown and conserved by micropropagation because of high coefficient of multiplication and small demands of the on number of initial source plants as well as the space. Here is the various techniques of plant cell and tissue cultures. The left top one is the callus culture, and below is the protoplast, and top right is the cell suspension culture, and top bottom right bottom is the anther culture. Nowadays, tissue culture is being highly practiced in agriculture. It allows the production and propagation of genetically homogeneous disease-free plant material. And the cell and tissue in vitro culture is a useful tool for the induction of somaclonal variation. The genetic variability induced in tissue culture could be used as a source of variability to obtain new stable genotypes, as well as the genetic engineering can make possible a number of improved crop varieties with high yield potential and resistance against pest. Genetic transformation technology relies on the technical aspects of plant tissue culture and molecular biology for production of improved crop varieties, for production of disease-free plant, virus-free plant, for genetic transformation, for production of secondary metabolites, for production of stress, salinity, drought, heat tolerant varieties. This is about the germplasm conservation. In vitro cell and organ culture offers an alternative source for the conservation of endangered genotypes, endangered plants, varieties. Tissue culture protocols can be used for preservation of vegetative tissue when the targets for conservation are clones instead of seeds, to keep the genetic background of a crop and to avoid the loss of the conserved patrimony due to natural disasters, maybe biotic or abiotic. The plant species which do not produce seeds, the sterile plants, or which have recalcitrant seeds that cannot be stored for a long period of time, can successfully be preserved via in vitro techniques for the maintenance of gene banks. And the cryopreservation plays a vital role in the long-term in vitro conservation of essential biological materials and genetic resources. Here is the plant propagation and germplasm conservation of Musa, Manihot, Piper, Cedrela, the economically important plants. This is about the protoplast isolation and somatic hybridization. Somatic hybridization involves the fusion of protoplast of two different genomes, followed by the selection of desired somatic hybrid cells and subsequent regeneration of hybrid plants. The protoplast fusion provides an efficient means of gene transfer with desired trait from one species to another and has an increasing impact on crop improvement. And they produce these are produced by somatic hybrids have been produced by fusion of protoplast from rice and beans using electrofusion treatment for salt tolerance. Here is the somatic hybrids produced by asymmetric protoplast fusion between Musa silk and Musa acuminata. A and B are the two different protoplasts. They have been hybridized. The consecutive steps from C to Z and H is the multiplication, callus, embryo, plants, potted, transferred to field. Here is a few examples of intergenic protoplast fusion. Raphanus sativus with Brassica oleracea protoplast fusion, making Raphanobrassica, Arabidopsis thaliana with Brassica campestris, producing Arabidobrassica, Solanum tuberosum with Lycopersicum esculentum, producing Solanopersicon, Nicotiana tabacum with Lycopersicum esculentum, producing Nicosiopersicon. All are the somatic hybrids, new genus, new genomes. Regarding somaclonal variation, Larkin and Scowcroft in 1981 proposed the term somaclone to describe the plants originating from any type of tissue culture. The genetic variation found to occur between somaclones in vitro system, which has been referred by Larkin and Scowcroft as somaclonal variation. The variation includes aneuploids, sterile plants, morphological variants, sometimes involving traits of economic importance in case of crop plants. The Ono variety of sugarcane is a Fiji-resistant somaclone. A sweet potato cultivar, the scarlet, was selected from shoot tip culture derived clone. Here are the three somaclones, Carina, Rebecca, and Sarah, from the camellia of Dipherbachia. These are the three different somaclones, somaclonal variants obtained from camellia. Here is the list of target traits selected by somaclonal variation in different crops. You can see rice, wheat, potato, sugarcane, sweet potato, tobacco, maize, and many others, where the different desired traits are being produced as somaclone. This is a case study with olive, micropropagated olive crop, olive orchard. This is the micropropagated olive orchard, and the olive oil extracted by micropropagated plants. Next is the, in tissue culture, in vitro culture, is the genetic transformation, which is the most recent aspect of plant cell and tissue culture that provides the mean of transfer of genes with desirable trait into the host plants and the recovery of the transgenic plants with modified traits. The technique has a great potential of genetic improvement of various crop plants by integrating in plant biotechnology and breeding programs. It has a promising role for the introduction of agronomically important traits such as increased yield, better quality, enhanced resistance to pests and diseases. And this genetic transformation in plants can be achieved by either vector mediated, what we call indirect gene transfer, or vectorless, without vector, what we call the direct gene transfer. The agrobacterium mediated genetic transformation is most widely used for the expression of foreign gene in plants. Successful introduction of agronomic traits in plants achieved by using root explants for the genetic transformation. And virus-based vector offers an alternative way of stable and rapid transient protein expression in plants. Transgenic plants of Jatropha were obtained by direct DNA delivery to mature seed-derived abaces via particle bombardment method. This technology has an important impact in the reduction of toxic substances in seeds, thus overcoming the obstacle of seed utilization in various industrial sectors. And another one, the marker-free transgenic plants of Petunia hybrida were produced using multi-auto transformation vector system. The plants exhibited high level of resistance to botrytis cinerea, the causal agent of grey mold. Here is some genetically modified crops under large scale cultivation: soybean, maize, cotton, alfalfa, papaya, tomato. They are transgenic traits and the transgene. I think I am going to be late. I should be hurried more. Okay. Okay. This is the last part of the seminar. The third one, the phytochemistry of plants. Phytochemicals are biologically active, naturally occurring chemical compounds found in plants, which protect plants from disease and damage and contribute to the plant's color, aroma, and flavor. These phytochemicals protect plant cells from environmental hazards too, such as pollution, stress, drought, UV exposure, or from pathogenic attack. And till now, more than 4,000 phytochemicals have been catalogued and are classified by protective function, by physical characterization, or by chemical characterization. And about 150 phytochemicals have been studied in details. And they are mostly secondary metabolites that accumulate in different parts of the plants, in root, in stem, in leaf, in flower, in fruit, in seed, and most of them are pigment molecules, which are often concentrated in the outer layers of the various plant tissues. These are the different types of phytochemicals in plants: phenolics, alkaloids, terpenoids, steroids. This is the origin and biochemical pathways of the phytochemicals, the secondary metabolites, which originate from the common precursors as products of primary metabolism. Three basic metabolic processes governed by photosynthesis, that is nitrogen metabolism, fatty acid metabolism, and carbohydrate metabolism, are responsible for the synthesis of secondary metabolites, like, as you see here, the alkaloids, terpenes, polyketides, flavonoids, quinones, coumarins, and are being used in defense against herbivores, fungi, bacteria, virus, other plants. They use it for the protection against physical effects of ultraviolet light, evaporation, chill, cold, as well as for attraction and stimulation, enforcing pollination, seed dispersal, oviposition, food plant sequestration, pharmacology, as well as for symbiosis. Here is the example of some secondary metabolites and their source plants. Few examples. This is the brief summary of various extraction methods for the natural products, like maceration, percolation, decoction, reflux extraction, succinate extraction, pressurized liquid extraction, supercritical fluid extraction, and the for respective method, the solvents used, the temperature to be maintained, the pressure to be maintained, the time required, and the volume of organic solvent required, and the polarity of the natural products extracted. This is the continued table: ultrasonic assisted extraction, microwave assisted extraction, pulsed electric field extraction, enzyme assisted extraction, hydrodistillation and steam distillation. These are the phytochemicals in medicinal plants. Their classification as a non-starch polysaccharide, antibacterial or antifungal, antioxidants, anti-cancer, detoxifying agent, and the group of compounds belonging to these classes are tabled and their biological functions. Different phytochemicals in medicinal plants with their biological function. Yes, this is the impact, a curated database of Indian medicinal plants, phytochemistry, and therapeutics. This is a comprehensive online database on the phytochemistry of Indian medicinal plants, the impact, which enables computational approaches towards natural product-based drug discovery. This database comprises of 1,742 Indian medicinal plants, 9,596 phytochemicals, and 1,124 therapeutic uses, spanning 27,074 plant phytochemical associations and 11,514 plant therapeutic associations. Here is the database and impact. Number of Indian medicinal plant, number of phytochemicals, the different type of associations, plant phytochemical association, plant therapeutic use association, plant medicinal formulation association, phytochemical human target protein association, plant pet phytochemical association, and the additional features. Thank you very much. Thank you everybody for your patience hearing. I am already 30 minutes late for the for my next meeting. Sorry, being make you delayed. I myself has also been delayed. Okay. Thank you very much. My God, did it you?
শোনা যাচ্ছে না, না শোনা যাচ্ছে না।
Hello, audible?
হ্যাঁ, এখন শোনা যাচ্ছে।
Professor Kaur just delivered his keynote speech. Thank you, sir.
Thank you.
Excellent. Very nice and vibrant speech, strong enough to inspire us. Professor Kaur provided valuable information regarding how cytogenetics entered the molecular era with the introduction of in situ hybridization. He emphasized on the use of site-specific probes in fish technology, and this will enable us to map chromosome rearrangement. India is a country of great biodiversity. We have natural resources. We have enormous potential. Professor Kaur discussed the importance of natural resources and their application in food and health. He also highlighted the power of tissue culture and genetic transformation technique for large-scale propagation of plants and development or generation of genetically modified plants for production of metabolized phytochemicals and for development of high-yielding crops, relevance to industry for commercial application. Thank you, sir. Thank you so much.
Thank you very much. Thank you. May I take your permission to leave? I have had another meeting at 12. May I take your permission, please? Thank you very much. অনিতা দিকে দেখতে পাচ্ছি, অনিতা দিকে
Thank
দেখতে
you, sir.
পাচ্ছি। হাই হাই অনিতা দি, থ্যাংক ইউ থ্যাংক ইউ থ্যাংক ইউ থ্যাংক ইউ
Thank you very much, sir.
Thank you, sir.
দীপক দা খুব সুন্দর হয়েছে স্যার।
ও আচ্ছা, থ্যাংক ইউ থ্যাংক
খুব
ইউ
ভালো,
থ্যাংক ইউ
দারুণ, খুব ভালো
Thank you.
বলেছেন, গুছিয়ে তিনটে ব্রাঞ্চকে কভার করে খুব ভালো হয়েছে, আমাদের খুব ভালো লেগেছে।
থ্যাংক ইউ
অনেক
প্রফেসর।
ইনফরমেশন পেলাম দীপক।
ও হাইসি বাবা, আপনাদের কাছে ইনফরমেশন, ইউ আর লোড অফ ইনফো, ইউ হ্যাভ উইথ ইউ দ্য লোড অফ ইনফরমেশন, লোডস অফ ইনফরমেশন। থ্যাংক ইউ অনিতা দি, অনেকদিন পরে দেখতে পেলাম। নির্মাল্যদা, অনিতা দি, সবাইকে দেখতে পেলাম, খুব ভালো লাগলো। থ্যাংক ইউ প্রফেসর মন্ডল। I take your permission to leave because I have a meeting started at 12. Okay, thank you.
Okay, thank you, Deepak.
Thank you, Nirmaloda. Thank you.
Thank you. Thank you.
Thank you.
Thank you, our HOD sir. Moving to the plenary lecture session 1. Now I would like to Professor Subrata Mondol, Department of Botany, Visva-Bharati, and Dr. Shakuntala Ghorai, Department of Microbiology, Raidighi College, to chair this plenary session 1. Sir, please.
So, am I audible?
Yes, sir, you are
Yes, sir,
audible.
you are audible.
Okay, namaskar. So we are going to start the plenary session, plenary lecture 1, that is first plenary lecture, and I am thankful to the organizer for giving me the opportunity to chair this session along with Dr. Shakuntala Ghorai of Raidighi College. It is my pleasure to introduce Professor Anita Mukherjee. Professor Mukherjee is well known to us and help us. Am I audible? Hello.
Yes, sir, you are
Yes, sir.
audible.
Yes, you are audible.
Professor Mukherjee is well known to us and helps our department in various ways. Her research interest in the field of nanotoxicology, phytoremediation, and biomarkers for assessing genotoxicity of anthropogenic compounds. And like this,
শোনা যাচ্ছে না কিছু তো?
সুব্রতদা,
স্যার আপনাকে শোনা যাচ্ছে না কিছু।
আমার মনে হয় কি অনিতা দি স্লাইড শেয়ারিং করছেন বলে কি ডিস্টার্ব হচ্ছে নাকি?
অডিওটাতে তো ভাবে হওয়ার কথা না।
আনমিউট, এই আনমিউট, আমি তো নিজে করতে পারবো, আমারটা শেয়ার করতে হবে না, তোমরা শুধু আমাকে পারমিট করো শেয়ার করতে।
hello, am I audible?
And now you are audible.
Now you are audible, sir.
For long time you have been
More
audible.
than, she published more than 150 research papers in national and international journal of reports and also different book chapters. She also produced 20 research students. And with these few words, I request Professor Mukherjee, our beloved Anita ji, to deliver his speech.
কোথায় চলে যাচ্ছি? I want to discuss. Thank you very much. I would like to thank the organizers for giving me this opportunity to present some of my work in presence of this august members. Now the topic of my lecture is today interaction of nanomaterials in planta, fate and behavior. Now we all know what are nanomaterials, but for the benefit of the students, I would like to say that Nanomaterials, they can be defined as natural, incidental, or manufactured materials containing particles in an unbound state or as an aggregate or agglomerate, where at least 50% or more of the particles have one or more external dimensions in the size range of 1 to 100 nanometers. Now, like all technology, any new technology has side effects. This nanotechnology has also its pits and falls. And it is interesting that with any new technology, then we must also know what are the threats it poses. Now, these production of the nanomaterials, they may be discharged into the air, they may enter the consumer product, and ultimately they may be either recycled or they may be disposed as a waste. They may reach the landfills and ultimately contaminate the water. Now, if you look at the literature, though this is a little bit older, till 2016, you can see that a lot of work is being carried on to know the toxicity of nanoparticles and also the genotoxicity of nanoparticles, which are arising, increasing at exponential levels. And if you look at the area of research, you will see the general toxicity versus the studies in plant.
Sorry to disturb you, ma'am. Slides are not changing.
Slides
are not. No.
Continue sharing.
Continue sharing. Where is, wait, resume sharing.
Yes.
Wait. Now, can you, okay.
Yeah, now continue.
A minute, sorry. So, can I go back? Can you see now?
No, ma'am.
No, ma'am.
1 minute. Can you see now?
The old screen is back now.
Don't put it on full screen. Never put it on full screen.
Can you see now?
Escape.
Okay, 1 minute. Okay, I won't put a full screen. Let me go. Resume. Resume.
Yes.
No, this is not showing.
It's okay. Continue.
কোথায় যাচ্ছে না তো এটা ফার্স্ট
ফুল স্ক্রিন করা যাবে না
আচ্ছা ফুল স্ক্রিন করলে বলছি ডিস্টার্ব হচ্ছে আচ্ছা let me go with the so this was the first one which was I was talking about the title of my work, followed by what are nanomaterials, which I mentioned that they need to be studied and how they are posing threat to our environment. And if you look at the studies which are being carried on so far, though this is a bit old one till 2016, you can see that the study of toxicity of nanoparticles and the genotoxicity are both increasing at exponential level. And if you focus on the area of research, that is the general toxicity versus the studies in plant are very negligible. But we know the plants are very important component of the ecosystem. They need to be studied and they mirror the ecotoxicity of nanomaterials, and they are the initial indicators for their potential biomagnification through our food chain. So therefore, the uptake and accumulation of nanomaterials within the plant systems cause large-scale ecological ramification. Now, is it audible? The plant systems which we had assessed in our laboratory include some in vitro studies and for our in vivo studies. For majority of the studies were carried for in vivo systems like Nicotiana tabacum, and the mother is calling Visva faba, Allium cepa, vis-a-vis some in vitro test systems like Fiscomitrella patents and tobacco BY2 cells were also studied. But today I am going to focus my study mainly on the in vivo systems which I have assessed, which we have carried. Now, a very important aspect of studying the nanomaterial is to do its characterization, either in single nanoparticle stage, Can you see it?
কেন যে এই হচ্ছিল গ্লোবাল
দেখা যাচ্ছে না? দেখা যাচ্ছে?
Yes, ma'am. Plant systems assessed. This slide is now visible.
Now characterization is visible?
No, ma'am.
No.
Okay, I will go to the individual then. Okay, now small one.
এখন দেখা যাচ্ছে, ঠিক আছে।
Now visible.
এটা কেন হচ্ছে গো? any দেখি anyway বাড়িয়ে দিই একটু, দাঁড়াও একটু ছোট করি।
হ্যাঁ, এটা ঠিক আছে, বড় হয়েছে।
হ্যাঁ, এটা ঠিক আছে। হ্যাঁ, so in studying nanomaterials, characterization of the particles which we are going to study is very important, whether they are as occurring as in a single stage, or because most of the experiments are carried, we dissolve in water or in media, so the colloidal stability as well as the morphological characterization is very important, which can be done through TEM, SEM, EDX, AFM of the single nanoparticles. Once we buy from the companies like Ulrich, Sigma, whatever, you have to do it once again in your lab again, using your resources, and to check the colloidal stability, the dynamic light scattering technique and zeta potential is also very important aspect. So this characterization is an important parameter of study which one should do at the beginning. And among the nanomaterials assessed, we have done quite a number of them, but here I am mentioning a few: the multi-wall carbon nanotubes, silver nanoparticles, nanoscale zero-valent iron particles, titanium dioxide, as well as zinc oxide, aluminium oxide, chromium oxide, gold nanoparticles, to name a few. So the first step, we have to do the adsorption. We have to see through SEM or through TEM, we have to see the uptake and accumulation, and EDX, which is very interesting that we have started I'm showing a study where we have studied the adsorption on allium cepa roots. As you can see in the upper panel, how the root tip of a control versus those of the nanomaterial, which is in this case is a zero-valent iron particle, how it has affected the root tip structures, the epidermis, the root hair, and also you can see the particles are adsorbed on the surface of these roots. Now, we mentioned that, okay, we understand that we can see that they are being these dots, white dots. Are they really the compound which we are measuring? So what you can see through SEM EDX in allium cepa, we found that in the control there was no presence of iron. Vis-a-vis in the treated set with 500 milligrams per liter of zero-valent iron in allium cepa, we see that yes, the same EDX shows the presence of iron. Now to go to the next one once again. And so what it does, it can cause immense damage. This adsorption, A is a normal root tip, B through SEM, you can see these white particles, the arrows are pointing, this is more enlarged, the more particles are clearly visible on the root tips of allium cepa, followed by the multi-wall carbon nanotube treatment. In the lower panel, what you can see, this is a normal cell, control cell, ultra-thin sections through TEM. You can see that normal versus a treated one with the MWCNT shows disorganization of the cell, the cytoplasm, the nuclear organelle, all have been affected. And you can see that there are large vacuolation. In plant, this is a very common effect that the large vacuoles are formed. The entire, as you can see that all of these, there's extensive loss of nuclear organization, ruptured plasma membrane, shrinkage of protoplast. At the same time, on the C, the particles which are shown here by arrow has been enlarged here, and you can see these black dots of carbon nanotubes. Next, similarly, in zinc oxide nanoparticles, the upper panel once again, it shows all the normal type of cells. This is a normal cell. You can see that it has a normal cellular organization, defined nuclear and organelle structures, whereas D, E, and F, it shows that extensive vacuolation with nuclear organizations lost, and there are also some ruptured plasma membrane as similar to that of the nanoscale zero valent ion. And further enlarging these particles, these black dots, so from this, they show prominency that they are there deposition of these nanoparticles of, in this case, it is zinc oxide nanoparticles, was clearly visible. Now, similarly, with silver nitrate nanoparticles, in the root tip cells, the D is a normal, which is a control, versus the E in allium cepa. Once again, we can see how this silver nanoparticle can cause damage, forming large vacuolation, disturbing nuclear organization, rupturing plasma membrane, like previous. So, to say in short that we have seen that the nanopart plants, they are taking up the nanoparticles. Mostly they may be in adsorbed, or they are also seen inside the structures of the root through the TEM studies. Now, since our laboratory is concentrated on the genotoxicity of the nanomaterials, we went to study the genotoxicity of nanoparticles. And we know from literature they can cause direct damage, they can cause indirect damage through ROS-mediated pathway. And these are some of the parameters which can be measured, and we have also done. We have measured the cytogenetic alterations through mitotic index, MN and CA, chromosomal aberration studies. And the DNA strand breaks were studied through COMET assay and DNA laddering. And ultimately, you can see that these nanogenotoxicity is definitely is a cause of concern. And initially, when we started, we studied the characteristics of the size, the polydispersity index, the zeta potential, and we found that they all had a role in the genotoxicity. And at the same time, such genotoxicity will definitely lead to a cascade of downstream effects, which will ultimately culminate in cell death. And this can be always, as I mentioned, that genotoxicity may arise through direct or indirect damage. Therefore, they can be studied by the chromosome at the nuclear aberrations, and we all know that they are important causes of concern, as they may lead to mutation and variations in polyploidy. One can do simple, we don't have much facility in our lab. So plain squash technique and staining by Orsin, you can see this is a typical anaphase stage where, and it is that all these arrows are showing you, the arrows will show you the black dots. These are black dots and nothing but the internalization of MWCNT. This internalization in a treated cell versus a normal cell. And the lower panel, you can see immense vacuolation has taken place. All these blue gap-like, bubble-like structures are nothing but vacuolations, and there are constricted dark stained nuclei, which is marked by the absence of nucleolus, which you can see in a normal. So this was clearly evident that MWCNT was internalized, and it causes induction of vacuolation in allium cepa cells. This is a more detailed, where general structures, some certain mitotic phases which have been studied, and it shows that we can there was formation of bridges, and also micronuclei, budding, and also there were, you can see some presence of micronucleus and formation of binucleate cells. All are results of the cytological changes induced by the multi-wall carbon nanotubes. And this, in another study with chromium, we could demonstrate elegantly using the ETBR stain. But with the ETBR stain, also the different stages of aberrations, the bridge, the micronucleus, etc., could be demonstrated as an effect of chromium oxide nanoparticles, and the system was allium cepa root tip cells. Besides that, one can use the DAPI stain, and study, because some of us have the privilege of what you call fluorescent microscopy, and one can see elegantly that how large micronuclei, binucleate, anaphase bridges can be formed, induced by a nanoparticle. In this case is a zero valent ion. Now this was all the mitotic stages, followed by the mitotic stages at the DNA level. What we did thus, we studied through Comet assay. We estimated the DNA strand breaks, and we saw that with increasing concentrations of MWCNT, there was increase in tail length of the comet, once again indicating that you could measure the effect at the DNA level. And this B panel, this again shows definitely we have isolated the DNA from the root tips and then carried the DNA laddering pattern, and you can see the bands corresponding inside have been highlighted in these red boxes. You can see the bands which show the breakage of the DNA as an effect of genotoxicity at the DNA level induced by MWCNT. Similar to MWCNT in Ileum-Cipa roots, studies were also carried in Nicotiana tabacum leaves, and you can say that these are the different concentrations selected, and these are the different types of comet assay measured. Suddenly, the initial stage 2 nanomolar shows much higher damage than the rest. One must keep in mind that these are not pure chemicals. So these discrepancies may be because of agglomeration, which is a natural phenomenon of the nanoparticles in solution. And to pass on to the next slide, which I'm going to show on the effect of similarly. Now, bulk versus the origin, the nanoparticle. In this case, zinc oxide nanoparticles was tested against parallelly with the bulk zinc oxide nanoparticles, and you can see the nanoparticle shows a greater DNA damage than its counterpart, the bulk one. Is same different effect and leaf of this quite obvious to the test compound, and therefore the root had taken up more damage and transferred into more comet DNA strand breaks, which have been plotted here as histogram. So with zinc oxide, now you have seen that the mitotic stages show different types of damages, and also the mitotic index also was different. So obviously it has some effect on cell cycle progression. It should have an effect on cell cycle progression, and therefore we carried some tests with flow cytometric estimation and showed that MWCNT induced alterations in cell cycle progression in Ileum-Cipa roots. And these are the histograms which have been plotted as a graphically. And to go on to the next one, now once this you have calculated that, now on the other aspect of the study was to measure methylation pattern. We can see the DNA methylation pattern also in Ileum-Cipa roots, which was studied. In this case, I'm citing only of this MWCNT, using two isoschizomers, MSP1 digestion and through HP1 digestion. This was a feature which we found, and we had used control versus PARC19 cells. And on the right one, to corroborate the similar effect, one can study the 5 MDC level concentration of the DNA in the root tip cells through HPLC, and it was definitely corroborating. The two results were corroborating each other. Now, initially I had mentioned that oxidative stress plays an important role in this DNA damage as an indirect mode. And oxidative stress not only causes an imbalance in the equilibrium between the ROS and the OX, at the same time, it also initiates the production of some antioxidant defense mechanism. We all know the H2O2, there's an upheaval of H2O2 giving rise to changes in CAT level, GPX, all the different type of enzymes, which are induced as a result of the stress. Simultaneously, there are also changes in some non-enzymatic systems. They are also affected by it. And this was tried out in experiments through root tip cells of allium, where we can see these are the different intensity of the ROS generation measured in our confocal using the fluorescent dye DCFTA. And by this intensity was transferred through a software known as ImageJ and plotted as histograms. And you can see with increasing concentrations, the ROS generation also increased. And this image is at a higher magnification where it is very clearly visible, these dots, the intensity, the dye, the DCFDA, how they are fluorescing at this concentration. That was by one MWCNT. This is by zinc oxide nanoparticles. Here also you can see that the intensity is increasing with the increasing concentrations. And another important aspect is that we can also study the mitochondrial depolarization. The mitochondrial depolarization was also affected because of ROS uphill. And what we see that on the upper panel is the localization of ROS by DCFDA staining. And in the lower panel, the rhodamine 1, 2, 3 showing mitochondrial dysfunction. This is a control where you can see orange dots, visible red with just a dot, a few more here at a higher concentration, and the highest concentration, it is totally absent. So to conclude is that this material like zero valent ion can induce ROS upheaval leading to mitochondrial depolarization. So going to the next one, like I mentioned that when ROS is induced, also at the same time, it is creating an effect on the antioxidant defense systems. And these are the defense systems which were carried. And you can see that in using zinc oxide nanoparticle, the ROS-mediated altered antioxidant enzyme activities in SEPA was measured. This is for H2O2 and MDA, GPOD, catalase, total thiol, protein thiol, and GSH. These are all studied in allium SEPA root tip cells. And this was studied, the oxidative stress response induced by aluminium oxide nanoparticles was also studied, where the nanoparticle versus the bulk was studied. And here that is the T-bar assay was calculated per fresh gram of leaves. Here also the H2O2 generated and the total protein content were studied. So what I want to say that we have seen that the roots at the plant system is capable of taking up or absorbing the nanoparticles, which is leading to cytotoxicity, genotoxicity, which could be measured and demonstrated in the earlier slides. And this nanoparticle-induced cytotoxicity can be manifested as necrosis and in programmed cell death. And so therefore, to study this, an array of vital stains and fluorescent dyes have been used to estimate them, and the mode of cell death was studied. And this one is the most simplest type. What you do, the root tips are subjected to your Evans blue stain, and the upper panel and the lower panel, they are subjected to the TTC, tetrazolium. And this one, the upper one, the dead cells which are permeable, membranes become permeable and they take up more blue color. And so the intensity of blue gradually increases, whereas TTC is just the opposite. It also, live cells will take up the color, that is the pink color, and the dead will gradually lose the color because they have lower mitochondrial dehydrogenase activity, and therefore they are going to exclude the TTC dye. So with very simple experiments, we can see that the mode of cell death, that cytotoxicity, how the percentage of cell deaths which are induced by the nanoparticles could be studied. And this is what I mentioned, that in ACPA versus VCFABA, the more the damage, the more the blue it becomes. That is the Evans blue. The percentage of dead cells could be measured as an absorbance of the color, and it is a measure of cytotoxicity. This was by zinc oxide nanoparticle. This is by zero valent iron particle, and all these are the simple root tips which can be could be photographed, and then you can also crush them and measure them, dissolve in alcohol, keep it for some time, and then it was quantitated and plotted against, and you can see that the dead cells that were induced in, and with the loss of permeability, and there was a reduction also in cellular metabolic activity. These are all telltale signs of cytotoxicity. And though in a mode of cell death, it is also very important and easy also to study the programmed cell death. So the material of interest was your nano zero valent iron, and in A, what you see, acridine orange, in this panel consists of acridine orange. Acridine orange, we know that it will stain the live cells into bright green. That is why you can see this entire intact root tip. The entire root tip is studied in confocal microscopy, and it is showing that with increasing concentrations, how this percentage of the live cells are decreasing from control 125 to 50 to 500. And the next panel, the ETBR, on the other hand, we know it stains the necrotic cells, which are stained orange. And this also, the intensity of orange is also increasing, corroborating, helping us to conclude that there are more dead cells now, more necrotic cells now with increasing concentration. If you can merge both, you can see both of them in this third panel. This was the intact root. This is the cells in your what you call isolated nuclei showing dual fluorescence. This is the acridine orange, this is ETBR, and these are the merged one. And this here, the live nuclei control. EA is early apoptotic green. Here is a mixture combination of yellow-green is the late apoptotic, and totally orange is your necrotic one. So programmed cell death could be also manifested by this nanomaterial in allium cepa root. And to come back to the summary of the work, I can say that these are the nanoparticles which have the capability of entering through the cell wall. They may be taken up inside, they pass the cell membrane, they may lead to genotoxicity, cytotoxicity, and the ROS generated in the mitochondrion will result into upheaval of certain enzymes, antioxidant enzymes like SOD, H2O2, GPOD, CAT, GSH. In our experiments, in general trend, we saw that GPOD got up, catalyst decreased, GSH also increased. And to sum up, the cytotoxicity, as you can see here, it can all lead to cell death and damage. So this was my final slides, and I would like to acknowledge my collaborators, Amitav Mukherjee and Momita Bandopadhyay. At the same time, my PhD students, Monoshid, Ilika, Urpita, and financial support from the CSIR, CRNN for the instrumental help, and UGC, of course. So thank you very much for your patient hearing, and I am ready to take a few questions if there are any. Thank you very much, the organizers.
Thank you so much, ma'am, Professor Mukherjee, for an elaborate and enlightening presentation. We really appreciate the way you have cited so many examples of interactions of nanomaterials with plant parts. and also their cytotoxic and genotoxic effects of different metal oxides like zinc, titanium, aluminum, and others. And ma'am, very lucidly, ma'am explained their effect on different cellular events like cell cycle, and also explaining the generation of oxidative stress, mitochondrial depolarization, and apoptosis as well. So if anyone have any question, you can actually unmute yourself and present that directly to ma'am.
It's okay, maybe it was clear enough so they don't have any questions. So I think I should...
Ma'am,
ma'am, I have a question.
Okay.
Okay.
Obinabu.
Obinabu, yes, ma'am.
Yes.
So, ma'am, this ZNO nanoparticle, ZNO nanoparticle has effect on Ross generation in Allium stipa roots, but with this increasing, but at the highest concentration of ZNO, there is a decrease in fluorescence intensity or the Ross generation. So why exactly does an organism like this Allium stipa decreases this Ross concentration in a higher concentration of nanoparticle?
It's a good question, Abhinavu, but one thing which I would like to always mention, that nanoparticles are not like the true chemical compounds. In true chemical compound, in any reaction, like with increasing concentration or decreasing concentration, they may be directly proportional, any parameter which you are measuring. Suppose Ross, if you take a pure compound, pure chemical compound, with increasing concentration, you will see a very nice curve, straight curve. But in the nanomaterial, what is happening? They are not, the colloidal stability will change. Yeah, that is why it maybe in 2 millimolar, it is showing the maximum. Maybe at that stage, because we have seen also that there is maximum, they are at the nano state, they are not in agglomerated, they are not in the nano form. Whereas at higher concentration, at 8 particularly, 8 millimolar, it has gone down. This is because, and we have checked with corresponding atomic absorption studies and all, and through TEM, that here the particles have agglomerated and they are no more in the nano state form. So there are some changes. They may be in nano, always the size will change. When you're buying from the supplier, they have given that this is of 10 to 15 nanometers. When you're dissolving in solution, that is when in every solution before doing experiment, you need to see the size through DLS and through TEM. I mean, am I clear? That is why the Ross.
Yes, ma'am. Yes, ma'am.
That is why the Ross we have seen at higher concentration. It was expected that there will be more intense.
More, yes.
No, but it has gone down. At a certain concentration, it was up, and then it was.
Because at higher concentration, the nanoparticles are not nano there.
Yeah, and they tend to agglomerate. They are not so easy penetrable. They are maybe in nano form, not exactly in 10, 20 or 100. They are much in bigger sizes. So penetration problem and accumulation uptake causes this change.
Okay, ma'am. Thank you, ma'am.
Thank you.
Ma'am, I have one lame question. Actually, cell does not know what is nano or whether that is synthetic or that is natural. If in the cellular milieu, actually during this agglomeration, elaborating Obinobo's question actually, that what will happen actually, whether cell will have any equilibrium between the synthetic and the natural particles available in the stressed condition?
The cell cannot identify actually which is synthetic and which is any foreign body will be taken up by the the cell, and it will be preserved in a vacuole. No, that is why vacuolation is studying of vacuole formation is very important for these nanomaterials. And the cell cannot recognize whether it is a synthetic or any foreign material. Whenever it enters the cell, it is stored in that vacuole. It goes straight into the vacuole. There only it is being. But here in the nanomaterials, they are also not only kept in the vacuoles, they are also disturbing the function of the nuclear function and the cellular function. As you can, I have shown in the slides through the TEM studies, you've seen how a single cell has withdrawn from the surface, and it has shrunk, and the protoplast and all, and plus particles like the nanoparticles could be easily visible. Is your question clear? Was it
Yes, ma'am. Yes, ma'am. Okay. Actually, I wanted to ask whether in sublethal concentration that will be stored in the vacuole itself. No.
Anything a plant. Tendency of a plant is to take up and keep it in the vacuole, and therefore there it will try to detoxify or get rid of it in whatever form. That takes time. It will either build up suitable enzymes for it to get rid of it. That is the natural.
Okay. Thank you. Thank you so much.
Thank you very much.
Ma'am, we are very happy to share a small token of appreciation here for you. I'm just presenting my screen, ma'am.
Yes.
This is for you, ma'am, and we humbly request you to kindly accept it with our sincere thanks.
Thank you very much. I'm so honored to be a part of your group in this COVID situation. You have done a wonderful job in organizing this, and I wish you great success for this. You have another day tomorrow. Thank you very much.
Thank you, ma'am. Thank you so much.
Thank you, Anita ji.
Thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you. Shall I?
Now? Yes, ma'am.
I will leave. I'll have my lunch and then join again. Okay.
Okay, ma'am.
Okay. Thank you, ma'am.
Thank you. Now it's time to start the second plenary lecture. I would like to request Professor Chaudhary Habibur Rahman, Department of Botany, Visva-Bharati, and Dr. Debashree Saha, Department of Chemistry, Raidighi College, to chair the session. Sir, please. Ma'am, please.
So may I start to introduce our honorable speaker of this plenary session, the second session?
Yes,
Okay, thank you. So it's my great pleasure to introduce my one of the best friend, Professor Tapun Kumar Maiti. Professor Tapun Kumar Maiti, he is the professor in the Department of Pharmaceutical Technology of Jadavpur University, and he was the former head of the Department of the Pharmaceutical Technology of the same university. His specialization in pharmaceutical chemistry. He is the eminent scientist in the field of the pharmaceutical chemistry, and also a very renowned teacher. So both accumulated in him, and in a very better way, or rather the expected way, it is modified his pedagogy as well as the curiosity of exploring source from the natural resources, mainly the resources subject, has been reflected during his master degree, period of master degree, Jadavpur University, by the award of the Professor Anupam Sengupta Memorial Bronze Medal. He is securing the highest mark in the pharmaceutical. He is having more than 30 years of research experience and also the teaching experience. Scholars and quite a good number of students are now working under his able supervision. He published so far 90 national as well as the international journals. Apart from that, he guided the M.Pharm and B.Pharm students, all things. He handled so many major research projects funded by UGC's CSR and other central and state level funding agencies. Sorry. He visited many countries like the USA, Germany, Austria, Switzerland, Netherlands, and others. And he have delivered so many scientific deliberation in the refreshers course, in the conferences and symposium, all these things. He is very much active member of the Board of Studies member, the PhD committee member, and the chairman of different governing bodies of so many pharmaceutical colleges and central universities and the state universities. And he is the member of the live member of the Indian Pharmaceutical Association. And apart from that, so many other scientific bodies also. With all these and editorial board member is also in so many journals, editorial board member. So with these words, I am requesting my best friend, Professor T.K. Maiti of Jadavpur University to deliver his speech. He will be talking on today the potential of plants' secondary metabolites as life-saving drugs, covering the chemoprospecting, gene prospecting, metabolomics, and definitely the pharmacological and clinical studies of so many biomolecules derived from the plant sources mainly. So with all these words, I'm requesting Professor Maiti, please.
Yes. Thank you, Professor Hussain, and good afternoon to everybody. I think all of the delegates, participants, speakers, resource persons, especially Professor Hussain, as he is my close friend. He has told so many things. I am a very simple man and having a very little knowledge. I may be a little bit odd man out in your scenario today. Because so far the plant is concerned, you are the God for that. We have to always take the help and advice of the people, those who are really working on plants. Something we do a little bit of fabrication works, as you said. So we shall be discussing today. I may not be that much of depth of knowledge to share on plant, but whatever the secondary metabolites do in the human health, that I will be highlighting something. So potential of plant secondary metabolites as life-saving drugs. So far, our history says that if about 125 or 130 years back, there were no synthetic drugs to save the life on ailing human life. I mean, all of you or some of you might be knowing that the son and daughters of Vishwakavi Rabindranath Thakur also died of cholera and malaria. Even Acharya Prafulla Chandra Rai also suffered a lot during his first visit to Edinburgh, whenever he had been there for his PhDs. But later on, he has obtained also DSc there. And he was suffering of so much of amoebiasis and gastric problem that in the seat, he was just lying in the seat whole the journey. I think somebody of you have got this information also. So just 100 or 120 years back, there was no such drugs to save the life, but we have enormous potentials of the drugs in the natural sources, particularly plants and microbes later on. Obviously, it explored later on. But plants, it was there, but as isolated molecule was a challenging job to have that time. People used to have the tincture, extract, and juices, and different kinds of traditional treatise people used to have. And as a result, in the advent of science, technology, I mean your chemistry, medicinal chemistry also came to the purview, then biochemistry, then microbiology, all these things have come, and we have come across a long way to have the modern drugs now. Though the mother drugs, that was the clue from ethnobotany and ethnopharmacological information, sometimes the folklore also, traditional system also has given some highlights regarding the department, regarding the activity of the drugs. So people have used it. So we shall be discussing details, I mean, just here. Importance of natural drugs, as that one quotation is there, that usual success rate of discovering new drugs from natural source. Microbiology, microbes are also huge potentials. Actually, that is another slides, because it is also a very big one, based on not only the conception but also on the implementation of ingenious comprehensive strategies, which invariably explore and exploit the untapped potential of natural resources. Now, we used to know that quinine has been used, not quinine, that cinchona bark juices or extracts have been used since long for different curing of some kind of fever. At that time, malaria was not the term to have that it is alleviating by cinchona plants and all these things, but it was there. And in the very beginning of the 20th century, huge work was on the cinchona was there, and you might be knowing that the Britishers, as they were visiting so much of remote places, also some of them were also suffering from different kinds of fever, and as a result, they realized the importance of cinchona. And in Darjeeling also district in West Bengal, that is in Mangpu, they have also a very good plantation. Cinchona plantations was there in 1856 or 57. They started extraction of that, but that time it was not isolated that quinine is there. But later on, the scientific innovations, a huge number of works have been done. I think cinchonine, cinchonidine, quinine, quinidine, a number of alkaloids have been isolated, and their potentials also have been reported. And when quinine was in hand, actually people got the life that malaria is no more uncurable. I mean, it can be cured by the quinine sulfate and quinine. This hydrochloride injections and all these things, but it was so much of bitter, and the production is also a question that how much of quinine as a pure drug is available to treat the malaria. So huge number of works has been done. But again, if somebody was to go on working on quinine, that is the D'Souza et al., whatever they have quoted here, that untapped potential which is yet to be explored. Thousands and thousands of plants have been explored, obviously, but thousands and thousands of plants also still today to be explored, right? Then similarly, the marine biology is yet to be explored, because collection and harvesting or collection and all these things are a challenging job. Whatever we see, particularly in the National Geographic Channel or Animal Planet or something like that, some research work is going on, but it is very difficult, particularly in our country, that going to the ocean bed and collecting the samples and then making them all preservations and all these things and do the work. But nowadays also there is huge scope is there that marine biology has to be explored properly, because there are so many cardiovascular drugs, anti-cancer drugs are also coming. Now, natural drugs substances offer 4 vital and appreciable roles in modern system of medicine, because the plant-based medicines, the plant-oriented plant exploited the drugs, that was the mother drugs, so to say. The synthetic medicinal chemistry developed a lot in 1930s or so. Organic chemistry also developed a lot later on, because organic chemistry earlier days, it was very much squeezing. People did not know. There was a idea that everything, that vital force theory, that is, it is God gifted, God is giving this, God is distributing this one. Whenever somebody is suffering from fever or sometimes of disorder or disease, people are using that. In earlier life, I mean, your past life, you are doing some sin things and some abusing things. That's why God is penalizing you. So this was the so-called, so you can say that myth, or people did not know. But with the development gradually, yes, there was a challenging job for the isolation, identification, and characterization or purification of the natural product, particularly from plant-based medicines or even from microbes. I am not going to touch microbe because it is huge again. Somebody of you of microbiology specializations are there. We have lots of good presentation also in microbiology. So morphine was the milestone drug, you can say. It is a narcotic analgesics. As a man or specialized in botany or taxonomy, you will be much knowing better that narcotic analgesics. So to say, analgesia was a term which alleviates the pain, right? But narcotic analgesics, they will be obviously reducing the pain, mild to moderate to severe pain. Together with that of there, they can produce some narcosis, addiction liabilities. Addiction syndrome also may be there. But this morphine, I will share that this morphine, once upon a time, it was the only saving drug, life-saving drug for the thousands and thousands of mothers, those who are going to deliver, etc. Because so much of pain, because there was no that such drugs at that time except morphine. So morphine is a alkaloid. It is a phenanthrene alkaloid obtained from Papaver somniferum. You know much better the posta, whatever we say, posto. So it is having a use in our day-to-day system, in our vegetables or in the curry preparations, in our food as a spices. It can be used. And the morphine again is having, as it is an analgesic, there is a margin of safety is very narrow. These drug is a poison in certain doses, and drug is a drug in certain doses. In lower doses, the alkaloids, these are alkaloids. The alkaloids are a very good drug. Yes, in higher doses, if it is there by mistake or intentionally, it is catastrophic. It is life-threatening. People will die. So morphine margin of safety was less. As a result, there was earlier days, there was no local anesthesia. Local anesthesia, I think it has started about 1960s, 70s or something like that. Earlier days, all general anesthesia were there. So as a result, calculations of that dose of the drug, how much of anesthesia has to be put, and how much of painkiller has to be put to a patient, it was a little bit of dilemma, because so much of micro level balance was not also available that time, I am telling. So as a result, this margin of safety of morphine was in a question, and so many people, they have died due to the morphine poisoning. So sometimes it was the term was that operation is successful, the patient is died due to morphine poisoning. So the same, another one plant is atropine, a constituent that is atropine is atropine alkaloids. It is obtained from Atropa belladonna. Belladonna atropine, it is a anticholinergic. It can protect our hearts also in different trauma, accidents, and even in third degree or high burn also in cases. Atropine is very much essential to save the life. Yes, morphine. Whenever morphine is having narcotic analgesic, synthetic drugs have come to replace morphine, because it is very difficult to have the narcotic drug continuously. So morphine, instead of morphine, the synthetic drugs came by mimicking the structure of morphine, that is pethidine. So pethidine, methadone, nalorphan, levolorphan, all these are synthetic drugs, but the mother drug was morphine, right? So atropine, as it is an anticholinergic and antispasmodics, it can relax the smooth muscles of the lungs, ureter, uterus, gallbladder, all these internal or smooth muscles. This atropine is very much essential. Okay, Atropa belladonna, it's a plant you might be knowing much better. That atropine is obtained from there, and this atropine, as their amount or availability is always a question, because maybe throughout the world the consumptions, if you consider India also, Asian countries also, kgs and kgs of morphine atropine is required. So it is very difficult. Scientists have changed the drugs, I mean, in synthesis. So this is scientists in the great sense, or in the positive sense, in optimistic way, that they are the great manipulator. So atropine is there. Then we have got propanthelin and so many other synthetic drugs. Now, ergotamine, ergot alkaloids, I think, Claviceps purpurea, I think, from the rye plant, from the ovaries of the rye plant, they are being isolated. So ergotamine is an alkaloid. It is an indole alkaloid, particularly. They are having a potentials that two physiological conditions of the would-be mother, I mean, the mother about to deliver the baby and after the delivering the baby or the fetus, that induction of labor pain. The ergotamine, it is induced labor pain where the mother is not getting sufficient of uterine contractions to get the babies out. So in that case, yes, this drug also induce the labor pain. Okay. So earlier days, it was there that when the normal delivery was there, that so much of wear and tear in the uterine canals and the wombs and all these things, and huge blood loss was being reported, and as a result, the mother used to die of hemophilia, right? And because blood loss, huge blood loss was there, and in the village, rural areas, there was no scope of blood transfusions, so people used to die. So this drug again, after the delivery, to arrest the hemorrhage, postpartum hemorrhage, the drug is having the effect. So two physiological conditions, the same drug is working in different way. Physostigmine, for Physostigma venenosum, I mean the plant where physostigmine, the semi-synthetic drug has come, neostigmine and all these things. The Physostigma venenosum, this drug is having used in the glaucoma and myasthenia gravis, eye problem, or you can say the cycloplasia, the accommodation of the eyeball is in a question where IOT, intraocular tensions. Sometimes people say I am getting headache, and it's not actually headache. It's an intraocular tension where the eyeball is getting some problem in the accommodation. It is in autonomic drugs, ANS, autonomic nervous system drugs. Then quinine. Quinine, as told that quinine in the same plant, quinine is there, and the same plant, quinidine is there. So quinine and quinidine, they are nothing stereochemically, they are only the levo and reductory isomers, but the therapeutic activity is totally different. But quinine is an anti-malarial. All of you know that quinine has been used anti-malarial, but as quinine production is a question that how much of pure quinine is available or to be isolated and identified or characterized, it is a challenging job. It is not that much possible. So scientists have manipulated the minimum pharmacophore, the basic structure of quinine, and they modified this structure to have synthetic drugs. So 1960s actually, that chloroquine, hydroxychloroquine, amodiaquine, pentaquine, santoquine, isopentaquine, hundreds of quinine, quinoline derivatives have come, which were also solving the purpose of quinine. So some oral active drugs also came, but not the natural product, but the mother drug was quinine. Another component of this plant, obviously there are 110 components that to be reported, but so much of sophistication in the isolation, identification and characterization techniques are there, but those chemicals, phytoconstituents or secondary metabolites are yet to be explored. I do not know that 110, and we have only isolated or reported about 6, 7 or 10 like that. So quinidine, another one alkaloid of the same plant, they are having what is that, cardiac arrhythmia, I mean in arrhythmia, anti-arrhythmic drug, where irregularity of the heartbeat is there. Patient may suffer from atrial flutter or atrial fibrillations, where chest pain may be there. In that case, yes, the quinidine is one of the best drugs. Then scopolamine, this is also an antispasmodic. It is an isomeric form of atropine with one ethylene oxide group extra. Ephedrine, all of you know that ephedrine, it is a CNS a respiratory stimulant. You can say that in the common cough and cold or syrups also it is there, and nowadays they are very much available in the market due to this pandemic again. And diosgenin from Dioscorea deltoidea, there are so many other species also that from Dioscorea. Diosgenin is, it is a glycoside which is obtained from Dioscorea deltoidea, a tuber. It is a glycoside which is having a use as an intermediate for the synthesis, because huge amount is available in the nature for the cultivations they can get, and from there the diosgen is isolated, and this diosgen is used as a used as a precursor for the synthesis of all the sex steroids. I mean estrogen, progesterone, testosterone, all these are there where from diosgen is the mother, you can say from there they can be isolated, they can be synthesized. So Pilocarpus javorandi, as it is a local use is there that people are using in a bald head, but somehow it is a news and people are also purchasing this one Pilocarpus javorandi hair oil and something like that, but they are having some other uses also. Now, what they can do, they provide basic compounds affording less toxic and more effective drug molecules. So don't be afraid of chemistry. Someone chemistry person is there already. He will be knowing much better than us. Actually, salicin, it is a discovery of 1899. You can be astonished that 1889, a German physician, his father was suffering from arthritic problem. At that time, maybe the term was not arthritic, but it was arthritic problem, and he was unable to move. And the person, as there was no such development that time, so he used to extract and Salix alba, the juices of Salix alba, and applying on his father, and gradually his father was curing somehow. So later on it was isolated, identified that salicin is the main component for there. And salicin, as it is a glycoside, this is the glycosidic linkage, somehow due to this breakdown of this bond and oxidation of this alcohol that is producing salicyl alcohol. Actually, it is not salicyl alcohol, now it is salicylic acid, right? So salicylic acid, scientists, what they have done, they detached this sugar molecule and they acetylated this OH. So this is COOH and this is OH. This OH is acetylated with acetyl chloride or acetic anhydride, and they are OCOCH3COOH. This is acetyl salicylic acid. What is in the name of aspirin or dispirin we get in the market. So far, this was the discovery of 1899. Just this is the only drug, so to say, in the market till today, 120 years. The synthetic drug, 121 years, the synthetic drug has crossed. It could not be, I mean, no synthetic drugs exist there so much of, but acetyl salicylic acid still today as a best non-narcotic analgesics, they are having good analgesic activity without the narcosis, right? So scientists have got a very good drug that it's a analgesic drug to reduce the body pain, and without this salicin, because salicin getting in kgs and kgs or quintal and quintal is not possible. But this aspirin or salicylic acid, because scientists are capable, because from benzene or benzoic acid, they can get salicylic acid, from salicylic acid, from phenol also can be synthesized, and they have acetylated. Only acetyl salicylic acid is dispirin or aspirin. Whenever this drug is active, I mean proved, scientists, they are the great manipulator. They mimicking the structure they synthesize, because acidic moiety they keeping in touch, and some homologs or some analog series they have prepared, and hundreds of non-narcotic analgesics came. That is ibuprofen, flurbiprofen, ibuprofen, naproxen. There are hundreds of drugs have come, but the first drug is salicylic acid, acetyl salicylic acid, but the mother drug is salicin. This is the importance that all the chemistry, actually plant chemistry is full of organic chemistry. To become a good chemistry in plant chemistry, one must have a thorough knowledge in organic chemistry. Otherwise, it is very difficult. So, because all the secondary metabolites, the pathway, as you know, that acetate pathway hypothesis, cyclimic acid hypothesis, even amino acid metabolism, whatever they were coming for alkaloids and etc., all are organic chemistry. Plant is living organism. So, organic chemistry fully there. Exploitation of biologically active prototype towards newer and better synthetic drugs. So, everybody knows cocaine. It is a tropine alkaloid like atropine, but cocaine is from erythroxyl and coca. It is having a local anesthetic activity, but it is also simultaneously having a narcotic activity, right? Some addictions may be there. I think those were my contemporaries, Professor Mandal and some people are there. Maybe that in 1990 FIFA World Cup, Maradona was being banned due to using cocaine for his common cough and cold or some painkiller like that he used to have. In dope testing or some other testing, he was being found and caught hold, and he has been banned from the match. Otherwise, Argentina could leave another one World Cup that time. Anyhow, this cocaine mimicking the structure that benzoyl, this is benzoyl with ketone groups, benzoyl. So benzoyl oxy group keeping it the minimum pharmacophore in the para position amino group and diethyl methyl para amino benzoate. They have taken it. They have synthesized procaine. So cocaine is the natural product obtained from erythroxylon coca. From there, procaine has been synthesized. It is a synthetic compound, mimicking the structure. Scientists, as I told, that they can see what is happening there and which is the minimum pharmacophore, making some structural orientations they have synthesized, and they found that procaine is also a very good local anesthetics. So when procaine clicked in the market, then procaine, tetracaine, benzocaine, lignocaine, xylocaine, alpha-eucaine, bucaine, hundreds of canes have come. These are all local anesthetics. So though the mother drug, we are indebted to the mother drug is cocaine, but mimicking the structure, scientists have synthesized hundreds of local anesthetics, so which are being used by the common people, because cocaine getting is very difficult. Obviously, cocaine, as it is having some narcotic activity, it is also under the control of the Narcotic Control Bureau and all these things, NACO. Okay, so this is not possible. So people are synthesizing this one, and we got it. Modification of inactive natural products by suitable biological and chemical means into potent drug. Just whatever I told, that is saponin, it is steroidal. It is a steroidal compound. In our +2 science, we have little knowledge on steroids, cyclopentanoperhydrophenanthrene nucleus, but several synthetic routes, they will be giving you progesterone. So in cancer cases or in the contraceptives, in different types of uses are there of the sex steroids, but the earlier or precursor is diazoxide. Otherwise, synthesizing this one or still the steroidal nucleus is also a very hectic job. I mean, so many steps has to be performed for that one. Whatever the 4 ways we have mentioned here, there may be isolation of novel genotypes from marine and terrestrial ecosystems, that genotypes and all these things. Madam also told so many things. I don't understand that much, but I have little just overheard some things. Genetic engineering, creating novel and altered genotypes. So far, the genetic engineering, I think in the plant biotechnology, whatever we have got, success is that they are in the plant biotechnology, the production of the crops, food crops particularly. I think we are indebted to Professor, your G.V. Pant, Gopi Ballabh Pant, who is the pioneer man to establish the your ICAR, I think, these are Council of Agricultural Research. So this person and G.V. Pant, that university also, they do huge research work. And as a result, the rice we are getting now, the tomato we are getting now, because earlier the tomato, cauliflower or cabbage, these were the seasonal fruits and vegetables. But nowadays there is no seasonal. Everything is, every time it is possible. This is the gift of your biotechnology, you can see. So far the plant biotechnology, for the fruits and vegetables or harvesting items, it is well good. But unfortunately, this biotechnology approach or biochemical manipulation of the selected pathways could not increase the plant secondary metabolites. My discussion is that if could be that atropine could be biosynthesized kgs and kgs, or your morphine could be biosynthesized kgs and kgs, like that. That could not be somehow in near future. Maybe that rigorous research and advanced research may be available. In later on we can get. Biochemical manipulation of selected pathways, if it is produced by radioligand assays, people are also trying. Super sensitive and specific selection techniques and evaluation of varied bioactivities. Yes, nowadays, drug discovery scenario, earlier it was there that 15 to 20 years of rigorous research is going on, at least some thousands and thousands, crores of rupees involvement, 300 scientists potential, 1,000 patients. All these things having some from clinical trial, phase 1, phase 2, and phase 3. And after phase 3, only the drug will be marketed if it is satisfied all the protocols. Suppose last pandemic, whatever we are observing, not last, still it is going on, that we are all have taken the vaccines, it is in phase 3 clinical trial. So it is not yet actually, so far the protocol is concerned. So we are taking the drugs, vaccines, it is in phase 3. It was written, I think, if you see the vials, where it is written in the ampoules, I think, vials, that phase 3 clinical trial and effective only 60 to 70% and all these things. That means it could be much more time to take, but as urgency is there, necessity knows no law. So something we have used also. Now, what are the pioneer countries, those who are having some extracts, having in good stock? That is India having opium straw extract, but it is also a hide and seek game. You might be knowing that excise and the customs, they also don't leave them. A narcotic control bureau is there to control them, that only openly you cannot make it. Some people are making it in disguise, so that peddling is going on, drug peddling also like that. China has Artemisia annua. It's another life-saving drug. Artemisinin is the alkaloid which has come with the development in that quinine and quinine and chloroquine. All these malarial drugs, anti-malarial drugs become resistant, and maybe about 15 years back, there was a fever called cerebral malaria or a brain malaria. Within 24 hours or so, the person suffering from fever was collapsed, and it was a malignant malaria, and it was not being cured by chloroquine or something, this type of anti-malarial drugs. So artemisinin alkaloids have been from Artemisia annua, has been isolated, and Japan scientist, I think Yuto, who has been awarded Nobel Prize in 2016, I think, for this discovery of the artemisinin alkaloid for falciparum malaria. Now, USA ginkgo biloba extract, they are having ginkgo biloba, zincolide A, B, C, D. There are some glycosides which is used for vigor and strength. In Japan, Panax ginseng and Siberian ginseng. Ginseng is also used for the, particularly the old age people, for the vigor and strength. Madagascar, they are pioneer in Catharanthus roseus, where your Catharanthus roseus, vincristine and vinblastine. Even in Calcutta University, huge work was there in botany department, I think also. That vinolin, they have done a good work. Vinca rosea, it is a plant which is ornamental also, and in lowland also it is there, wetland also it is being available, and in the garden also it is just like wild also it is there, wild also it is there. So Eastern Europe, the ergot mechanical eye plants of selected fungus, that Clavisus purpurea, I told already. So this is the methods how the drugs can be obtained from the medicinal plants. So it is a very long process. Time is very short. We shall be going. The morphine is the narcotic analgesics, but another one, scientists manipulated the drug, that hydroxy group of morphine has been methylated. Me means methyl in organic chemistry. So same structure is there. Only one methyl group has been changed. It is semisynthesis derivative. So it is called codeine. So codeine is a antitussive. Codeine phosphate, all of the syrup, we know that in respiratory problem, when the congestions are there, or some tingling sensation in the throat regions are there, some antitussive drugs, it is there, that codeine is there. But codeine, as it is semisynthetic though, but morphine is the basic moiety. So codeine in kgs and kgs or tons and tons is difficult to get. People are synthesizing guaiacol, methorphan, dextromethorphan, these kind of drugs. Papaverine, the same plant Papaver somniferum contain another one alkaloid. It is called benzyl. This is isoquinoline nucleus, benzyl isoquinoline papaverine. It's a very good antispasmodic drug, or to reduce the smooth muscle relaxation, to have the smooth muscle relaxation activity, and the antispasmodic pain in the smooth muscles, abdominal pain or cramps, something is there. But as they are in natural product, availability is always a question. So scientists have synthesized so many synthetic drug, that is propantherine bromide, dicyclomine hydrochloride, and your penthionate. These kind of drugs have come. They have replaced, but the mother drug or the information that came from papaverine. Purine alkaloid, every day it is our cup of tea that every day we get some CNS stimulations. But caffeine is also, it is a xanthine of purine alkaloid. Caffeine is also a very good drug, it's a supplement as a diuretic. Those who are suffering from high blood pressure particularly, or having some edema, soiling of the legs and some regions, so that the increase of blood pressure will be there. So they can take caffeine, coffee also, or coffee arabica or Camellia sinensis, to have the urinations as a diuretic. It's a diuretics. Then theophylline, the same drug, having one methyl group less. It is 1,3,7-trimethyl xanthine is caffeine. This is 1,3-dimethyl xanthine is theophylline. This is a drug for bronchodilator. CNS stimulants, they are having activity, but their bronchioles, in the bronchospasms or even in the asthmatic conditions particularly, they can be used as a bronchodilator. Then cardiac glycoside, the term itself has changed the history of medicine and medical treatment, because everybody is hankering that high blood pressure to be reduced. But whenever the blood pressure is reduced to some extent, then also patient collapsed. Then how to tackle those situations? Nature is great. They are having dioxin from digitalis purpurea and digitalis lanata, a lanatoside or purpurea glycosides. So they can be used in the treatment of congestive heart failure, where the cardiac contractility of the muscles, cardiac muscles contractility is less, the output is less, and the blood pressure is too much fall. So in that cases, the dioxin, a drug, a cardiac glycosides, a steroidal glycosides obtained from a plant, digitalis purpurea. You know much better for identification of the plant. And digitoxin, another one, dioxin, digitoxin. Only there is some structural orientations. I will not go in details because here is one hydroxyl group extra there. Here is no hydroxyl group here. And here is another hydroxyl. Only one hydroxyl group has changed. That is, maybe that scientist has made it. And congestive heart failure, they can be also used. Reserpine in long run, some research is there. Reserpine Rauwolfia serpentina is a snake repellent in the traditional use, that in the courtyard of the house, if you put or cultivate this plant, maybe that snakes will not come. It's an indole alkaloid. Snakes will not be coming, and that was the traditional use. But later on, it was observed that it is a very antipsychotic drugs. In the mental illness, it can be used, but later on again, it has been found in 1940 or so that it can be used as an antihypertensive drug. So reserpine is an antihypertensive drug, but the thing is, as these are from natural compound, and you see the structure, you need not go the tidbit, just you see the structure, that this much of structure to be synthesized in the laboratory, it's a challenging job for the medicinal chemist, right? So as a result, scientists have manipulated, no, this structure, okay, keeping some pharmacophore, they have captopril, enalapril, this kind of drug, so that they have alprazolam and this kind of drug, so that guanethidine, then there are hundreds of drugs are there, synthetic drugs and antihypertensives. Then curer, DTO curarine, you have heard the name Strychnoxa castanei or Toxifera. There are the plant which is a curer. Actually, curer is having high level, it is poisoning, right? Curer poisoning, you might be knowing that in Sherlock Holmes, the story are there where the injections of curarine lead to the death of the people, and it was very puzzled for Sherlock Holmes by Arthur Conan Doyle that to get the persons identified, I mean culprit. So it was a challenging job for him. So ultimately, anyhow, you can get in Kiriti Omnibus also in Niharanjan Gupta in our Bengali who writes detective books also. There are some similarity because in the foreign background, some stories have been also written. They have given. Anyhow, this drug is in strychnine poisoning also can be used, in neuropsychiatry also can be used, and adjunct to electroshock treatment of neuropsychiatry. When the patient is having, so to say, mad or psychological disorders are too high, in that cases sometimes can be used. So vincristine and vinblastine, as I told, that these are anti-cancer drug, particularly ovarian and breast cancer, they can be used. This is the structure. The plant is only maybe half meter height, but the structure is also more than half meter in length, right? So this is the things that nature is great. Nature is silently synthesizing and biosynthesizing so many things, but unfortunately, plant is not doing the biosynthesis secondary metabolites for us. But fortunately, those metabolites are very useful for our different disease or disorders. So plant may be having that microgram or milligram level in their system for their survival from different adverse situations. But fortunately, scientists explore that those secondary metabolites are very much useful in different disease or disorders. Then vincristine, vinblastine, vindolin, these are there. It's an indole alkaloid. Another one, Taxol or paclitaxel. It's a very big story is there in America. There was huge trial and on this drug, and ultimately they came up with the success that they can be used against cancer. But this Taxol or paclitaxel, it said that a plant, or it takes about several years to become mature, and 3,800 mature trees has to be chopped out, or you can take the barks can be peeled out from the plant, and to have that some kgs of 8 kg or 10 kgs of Taxol. So how much Taxol will be costly, just to think of. If you consider that 5 years is the maturity of the plant to have the barks to be depilled from the stem. Now those barks has to be utilized for the extraction, isolation, and identification and characterization of the taxol or paclitaxel. So it's a very, very difficult job. That is why scientists have done your cyclophosphamide, meclorithamine, busulfan, melphalan, these kind of synthetic drugs, not the natural product. But it is also having a very much of use in the cancer. Whenever you can afford the money, I mean, for the treatment, when the life and death comes, yes, that can be used like Taxol. It is one of the best drugs, I think, in 21st century. I think this is one of the best anti-cancer drug. Obviously, some anti-cancer drugs from antibiotic scenario from doxorubicin and daunorubicin are also there. Mitomycin C is also there. Methotrexate, we say. Some antibiotics are there, but plant-based medicine is Taxol or paclitaxels. Quinoline as an anti-malarial, there are so many story about it, because your Ronald Ross is the pioneer man who actually, how the malaria was transmitted, people did not know. My father, who actually expired in 90 years, he used to say that the malaria patient, when it will be there and dying will be there, people will not be going for burning the dead body also, because they know that if you go also, we shall be suffering from malaria. So this was a huge myth people did not know. But Sir Ronald Ross in Hyderabad as well as in Calcutta also, he did the research work, and ultimately he found out the malarial parasites life cycle, that how the vector, I mean your Anopheles mosquito, female Anopheles mosquito, carry the germs and bite the human being, and human beings suffer from the disease, and people used to die because there were huge informations are there. At the time is short, it cannot be elaborated here. But somehow he only discovered the life cycles, and he got the Nobel Prize for that one, that how malarial parasites are being infecting the human being. And quinine and quinidine, that I told that it is also used in cardiac arrhythmia. The same drugs, just you see the structure, a beta configuration and alpha configuration, it is beta and alpha. So only the orientation of the hydroxyl group about the asymmetric carbon atom or chiral center, we say, chemistry person expert is there, they will be knowing much better. So only the orientation of the hydroxyl groups or that beta levo and dextrorotatory isomers, their action is totally different, and other synthetic drugs have come. So these are the mother drugs. Artemisinin, that I told, that Artemisia annualin, a Chinese plant, chloroquine resistant, it can be used against the Plasmodium falciparum malaria, a sesquiterpene-lactone. Anthraquinone glycosides, though there are no such synthetic drugs, particularly still today we are indebted to the nature, because aloe vera, cascara, sagarella, jalap, colocin, podophyllum, all these things are there. They were having purgative or laxative activity, and the drugs are amodine or aloe amodine. It is dihydroxy, what is that, your anthraquinone glycosides. They are also anthraquinone, but these are free. Obviously, 1, 2, 3. If there is some sugar moiety, then it is glycosides. And these anthraquinone glycosides, so far it is, you can say, unbeatable, because senoside A, B, C, D, they are also having dianthrone molecule. So these are unbeatable. So far, the purgatives are their synthetic drug has not replaced these natural products so far. And senoside A, B, as I told, the dianthrone type compounds will be there. And here it is zincolide, that is USA, particularly the old age people, they don't think it is old age. So they enjoy the life, they are vigor, and they are very much good musculature or physical. So they maintain all these things, and they used to have some zincolide and ginsenoside. Nowadays in India also some abroad companies also putting these materials, not only isolated materials, it may be extract or concentrate may be there. Having some tonic as a stimulant, and some vigor and good health and all these things, you can say. Ohimbine and pilocarpine, that treatment of glaucoma, that Pilocarpus jaborandi, as I told you, local use is there, and for that one in the glaucoma also it is being used. And ohimbine, that is Paucinia ohimbi, that Rubiaceae family, treatment of importance in patients with vascular or diabetic problem. As you know that most of the diabetic patient in long run, they lose their sexual desire. I mean, there are some biochemical problems or phosphodiesterase problems are there. Maybe they will be losing their aphrodisiac. Tropine alkaloid, that atropine as local use is there, that in European, particularly in European culture, earlier days, Egyptian culture, the ladies particularly, they used to have instillations of the juice of this Atropa belladonna to make their eyeball much more clear and a little bit bigger in size. So they are very much handsome, they are very much beautiful to look, their height and their fair complexion, and if the eye becomes very much of big size, that is what not, it is very beautiful. So they to pose them in the party and cocktail dinners, etc. they used to have for showing their beauty. So anticholinergic, antispasmodic, even mydriatic. This mydriatic activity is a local use of atropine. Yes, it has been taken by the ophthalmologist or ophthalmic surgeon, even ophthalmologist, particularly eye specialist, so that whenever the power is there, eye people, they used to instill that 5 minutes or 10 minutes you wait after installation of atropine sulfate drops in the eyes, so that eyeball or people will be much more bigger in size to see, and they can easily calculate that retina, how much of power has to be given and all these things. So mydriatic activity, anticholinergic as well as antispasmodic. The drug in local use is different, mydriatic, and drug is parenteral use is different, anticholinergic or antispasmodic. Hyoscine and scopolamine, pre-anesthetic sedations, medication sometimes being given, intraocular stramonium, in motion sickness, particularly sometimes they are very much good drug in homeopathy. We do not know that much, but Hyoscine niger, your Datura stramonium and Hyoscine niger, they produce this hyoscine and scopolamine as in homeopathy. Some medicines are there also. They used to have. This is berberine on alkaloid, Berberis tinctoria. This is actually having some your diabetic as well as hepatoprotective activity. Heliotropium zillionicum, lupeol acetate. It is also a triterpenoids, tetracyclic triterpenoids. It is also having your sedative, hepatoprotective as well as this activity. Then Echinocarpus frutescens, lupeol and alpha amidine acetate. These are the triterpenoids compounds. So with this, I remain, because it is huge. Actually, time is very short. I think I have already taken some more time. So please, I am over. Professor Hussain.
Thank you so much, Professor Maiti. It has been a wonderful lecture. For myself, as I am from synthetic organic chemistry background, I found use and application of plant metabolites for drug synthesis a very relevant topic for my field of work. Thank you so much, sir. It has been a wonderful presentation.
Thank you, madam.
Thank you.
Others too, I believe, have found it very beneficial and relevant, especially in today's world, when we are desperately in need for more and more natural product drugs to fight different diseases.
Right.
One such disease has locked us up for 2 years.
Yes.
Yes. So we are in desperate need for newer drugs to fight various common and uncommon diseases. And if derived from plant metabolites, this will have fewer side effects. Thank you so much, sir, for such a lucid presentation.
Thank you.
It's time for interactive session.
Thank you. I'm sorry, Dr. Shah.
Yes.
Sorry for interference. I'm very sorry.
No, that's all right. That's all right. Please continue, sir.
So
please continue, Dr. Rahman.
So thank you, Professor Maiti, for your nice presentation. And thank you so much for giving us so much time and sharing your expertise with us in the field of the organic chemistry, basically the pharmaceutical chemistry, and the bioactive compounds and all these things. Thank you very much.
Thank you. Thank you.
It's time for the interactive session now. I would request all the participants, if you have any question, please unmute yourself and please do ask the question.
This is an excellent presentation by Professor Maiti. It is really nothing, just excellent. outstanding. These words are appropriate.
This is my pleasure. This is my pleasure. Because
and while I am listening this topic, just I went through my down memory lane when I was a student of MSc Botany and Professor SPSN was taking our classes, plant chemistry.
Right.
And I had come across these many of such compounds, and how can we determine the structure and all those things, and how such modifications can be converted into other compounds, and so all these things, and these compounds which you have mentioned here, and they with enormous their biological activities with human welfare and in human medicine. This is simply I have no adjective, no word to say about this, and so a lot of discussion can be done over this, and I do not know whether the organizers will allow us to interact in this manner. Anyway, so the other audience, they have a right to ask questions. So just I have expressed my feeling that it is simply outstanding.
Thank you. That is your greatness.
Thank you so much, Professor Mondol and Professor Maiti. Any other question coming up?
ম্যাডাম বলছি স্যার একটা কোশ্চেন ছিল, শোনা যাচ্ছে?
হ্যাঁ বলুন।
হ্যাঁ শোনা যাচ্ছে।
বলছি স্যার, পলিহার্বাল ফর্মুলেশন যেগুলো আমাদের প্রিভিয়াসলি যেগুলো ছিল বা এখনো আছে, তো সিঙ্গেল কম্পাউন্ড আইসোলেশন আর তার ইউজ আর পলিহার্বাল ফর্মুলেশন, এই দুটোর কন্ট্রাডিকশনে মানে পলিহার্বাল ফর্মুলেশন তো আমরা অভ্যস্ত, তো এটা পরপর অ্যাবলিশ হয়ে যাচ্ছে, মানে এটা বাদ হয়ে যাচ্ছে, কিন্তু এর ইম্পর্টেন্সটা যদি একটু বলেন এবং যদি মানে এটা সায়েন্টিফিক্যালি রিকগনাইজ করার পদ্ধতি যদি একটু বলেন।
Okay, actually polyherbal formulation, it was thousands and thousands years old, right? Actually, if you consider the Charaka Sanghita, Sustrata Sanghita, and our Ayurveda also, if you say, it was there because at that time only so-called the Kaviraj or Buddharaj, they used to collect some plants in different season and different time. So time of collection, those who are as some of you are very much well known, that when the plant has to be collected or when the plant has to be harvested, where which part of the plant will be active in this particular, either in the sunlight or in the dark or in the early morning or in the afternoon. So there are so many photochemical reactions also. Like biochemical reaction, there are so many photochemical reactions also, that this much is available this time. So-called the Buddharaj and Kaviraj, it was a family traditions. They used to, that was Shruti. You might be knowing that Shruti means the father will be dictating the knowledge to the son, and son will be next. Like generation, it was coming that they are Buddha. I mean, this title also came in maybe in that way, whatever we are knowing. As a result, they used to, by their vast experiences, they used to collect different plants in different time and mix up to remedy a particular disease. So polyherbal, maybe holistically they were giving some activity, but in the modern science, combination of so much of components together, it is a question, because whenever we are putting one or two drugs, there is some interactions, drug interactions, or compatibility. We are saying that compatibility maybe drug interactions may be good to produce, that is synergism or potentiation, or sometimes they can produce inhibitions. So whenever in pure drug, one or two drug, when they are combined, their compatibility structural interactions are being considered, that whether they are giving a good effect or not. You might be knowing that a Ceftriaxone tablet, earlier days it was there, pyrimethamine and sulfa methoxazole, these two tablets, Professor Mondol, Professor Hussain, they will be knowing much better that in diarrhea or in fever, you used to have Ceftriaxone tablet. It is a combination of two drugs. And these two drugs, whenever they are being combined, they are giving a 10-fold their activity when they are administering individually. So these are all pure synthetic drugs. Now, whenever a extract question comes, polyherbal means you are giving extract. Now, in a extract, first of all, the extract has to be freed from solvent. What is the extract generally? Water extract, you cannot keep long time. These are instantaneous preparations. You just extract today and you take it now only, just like your infusion, whatever you do in the home nowadays. So when polyherbal extract is being given, stability always will be a question. I think you are getting my point, because stabilizing the things, antioxidant, preservative, or antimicrobials, these has to be right there. Now, polyherbal means hundreds of components are there. You do not know which component is working or which is not. So this is one kind of, you can say, that our belief that herbal medicine will be doing this one or this one. And sometimes people are getting some effect, some people may not get the effect because they are not validated. So to say, the scientifically they are not validated. And it has been also observed that sometimes in herbal preparations, and whenever recent research is being done, that particular activity is abolished. It is not there. This is due to what I am telling, because whenever it was a family business, that is through orations or through transmission of knowledge from father to son to son, like that, what happened, all sons or all people will not be that much of intelligent, or you can say wise, to remember all the things, because that time we did not have the documentations. You might be knowing that last 100 years documentation also we do not have. We are unable to reproduce the 100 years documents. I think only Botanical Survey of India, I think they are having some documentations, but most of them are termites burned or spoiled by termites and all these things. Your specimen or herbarium, whatever you say. Once I had been there in maybe 1989, 90, I had been there, that 300 years old some herbarium was being collected and that was being preserved. Dr. Tapan Malik or something, Mandal, something was there. He was the curator, probably, or scientist. He helped me, and I observed that some British scientist, he has collected this plant from Kalahandi district of Odisha. Just you think of about 300 years back, what was the road situation, and people has gone there and collected. So it is very difficult to say that polyherbal formulation, yes, old age preparations, where the people are unable to withstand high level of the toxic or synthetic drugs, sometimes they are taking, but the cure rate is always a question. I cannot say it is not working, but cure rate is always a question. And when people are failing allopathy or some other medicine, sometimes they say, okay, let us try once, because we have tried a lot, like that. But whenever a cancer patient will be there or a diabetic patient will be there, they will say that if you take neem leaf, you will be curing from diabetes. Is it true? Then why people will not take neem leaf throughout the world? It's not like that, but somehow our tradition is very high. But as the advancement of science and technology is there in medical field, huge advancement is there. We have to believe in unique components or maximum 2-3 components which can be coherently active to a particular disease. It's very difficult. Polyherbal, when you will see that some herbal preparations are there nowadays in the medicine shop also, it is so much of stinking smell people are taking. You see, corolla juice, what does it mean? The intact corolla is available in the market. Why people are going for corolla juice to get rid of the diabetes? It is a nonsense things. But somehow people are convinced that if I take this corolla juice, just you think of corolla juice, they are protected. How much of antioxidant they have to give, how much of protective they have to put there, how much of antimicrobials, because water extract means huge amount of microbes will be attacking them. Then how can it be? So keeping those products intact for long run, it's always a question. That's it. I think I have made some light on this.
ঠিক আছে স্যার, মানে ব্যাপারটা হচ্ছে ওয়াইসালন 10 mg চলছিল ভাই কোমরের ব্যথার জন্য, ব্যাক পেনের জন্য। তো সেটা দুবার চলার পরে তাকে সারানো যাচ্ছিল না। ও এমটেক ইঞ্জিনিয়ার, কাজ করছিল, পাচ্ছিল না কোমরে ব্যথা। তো আমি একটা পলিহার্বাল ফর্মুলেশন বানিয়ে ক্যারোটোপিস বিগানেটা, ক্যারোটোপিস প্রোসেরা, রিসিনাস কমিউনিস, ইউফোরবিয়া থিমালিডিস, প্লেয়ার, এইগুলো দিয়ে একটা কম্বিনেশন বানিয়ে ঘি দিয়ে বানিয়ে দিয়েছিলাম। তো বানিয়ে দেওয়ার পরে স্যার মানে ম্যাজিকের মতো ভালো হয়ে গেছে, এবং আমার এক বন্ধুরও ভালো হয়ে গেছে। কিন্তু এখন কোশ্চেন হচ্ছে যে এতগুলো গাছের কম্বিনেশন থেকে প্রিপেয়ার করার পরে মেডিসিনটা বানালাম, রাইট, এই রিকগনিশন কি? কিছু রিকগনিশন নেই।
No, you cannot make. What is happening? You have to go for a patent first of all, and a huge investment, and you have to get the license from the DCGI, Drug Control General of India, that I have the formulation. Even you might be seeing that in the TV, some channel, in the home channel also. Sometimes it is showing that NIST, National Northeastern Institute of Science and Technology, I think it is Jorhat or Assam, somewhere it is there, and they are producing some balm for the knee joints pain and giving some
advertisement. You will see all the rural people are coming and some advertisements are there and they are getting some benefit. They are showing that people, those who are unable to work, even they are lying on the bed, now they are okay. So as government of India is already funding there, I think that could be much more in the purview of BCGI, so that at least a painkiller, you are taking brufen, paracetamol, gada khachi amra, tai na? So it is a temporary relief, because some are degenerative disorders. এই যে বাত বলুন, arthritis বলুন, এগুলো degenerative, এগুলো তো আর form করবে না, because our limb glands, it is already dry up. The chondroitin sulfate and the what is that limb that joining which lubricate the system, particularly our that joint of patella and all these things. So chondroitin sulfate and N-acetyl glucosamine, এটা হচ্ছে আমাদের inborn in our system, in our joints. They are generally lubricating the system so that we can work freely and all these things. But after certain time, or due to some other degenerative problems, their formation become less, or in certain ages also, after 60, 70 something, some people will be getting scared of that one, the scarcity of those lubricants. So that regeneration is not possible. It is only a your temporary relief, that some balms or some ointments you can apply and have some massages and all these things, it can be. But temporary, permanently relieving, it may be a question. But as you are telling that you have done it from yourself, I congratulate. Please, you that's fine. But you then apply in others also, that you see, because statistics will say that if out of 100%, at least there are some 60-70% cures the same problems, then the drug must be having some value, obviously. Otherwise, polyherbal, because how to get rid of the alcohols fast? আপনার কি আপনি
না আমি এক্সট্রাক্টটা জাস্ট হিট করে এক্সট্রাক্ট তৈরি করে ঘিতে অ্যাবজর্ব করানো।
As you have applied ghee, there is least possibility, because ghee is having good viscosity or density, which may not be, but there is every possibility that the extract after some days will be spoiled due to some microbes or fungal growth or something like that, right? চেষ্টা করুন, আপনি যেটা করছেন, I should not discourage you, but
না ঠিক আছে, এগুলো রিকগনিশন পদ্ধতি আমি জানতে চাইছিলাম, যদি কারো সাথে অ্যাটাচ করে যদি এগুলো ফর্মুলেশন
রাইট, এটাকে আপনাকে তাহলে ইয়ে করতে হবে, আইপি, ওটা গেল ইন্ডিয়ান প্যাটেন্ট অ্যাক্ট, আমাদের নিজাম প্যালেসে আছে, ডক্টর আগে ছিলেন উনি, এখন রিটায়ার্ড ডক্টর মৈত্র, আমার এক জুনিয়র আছে ধনঞ্জয় আছে এখন, ধনঞ্জয় পাল, ডক্টর ধনঞ্জয় পাল, ও না, ও ছেড়ে চলে গেছে। There are some office in Nizam Palace, Indian Patent Office, IP or something like that, in Nizam Palace, just opposite to Minto Park. You can have some approach that I have this one, I want to make it your what is that patent, so that at least if they allow, there are some paraphernalia. I mean, you have to apply this one, you have to then pay some fees for the government and etc., and they will be getting it, placing it somewhere, then some court, all these things will be there, and ultimately it may take some months to have, if at all it is there. But you have to protocols have to be designed, otherwise it will not be, because curing one person or your brother, it may not be giving that much of weightage.
Sir, the organizing committee.
Excuse me, thank you so much, Professor Panda and Professor Maiti. I'm afraid we are running short of time.
Yes.
I'm
so sorry to interrupt.
I want to say something. Hello. I am from Department of Zoology, sir.
Oh, sir, welcome. Rakesh da, welcome.
Thank you. Sir, you are discussing about the anti-arthritis ointment that developed by Nis Jorat. Actually, this was a collaborative project from the Visva-Bharati Zoology Department and the Northeast Institute of Science and Technology.
That is advertisement we are seeing in the...
So Professor Samir Bhattacharya was there, the PI.
I know.
We have also worked with this project. There is a big project from CSR. So it is from the herbal formulation, etc. So we have actually from isopulegol, that kind of that lemongrass, etc. We have extracted oil, and that is actually tested for anti-arthritis. Not rheumatoid arthritis, but in rheumatoid cases also it gives relief. So we have distributed the ointments in the Shantiniketan surrounding area also. and some commercialization has been also taken care of. And so regarding patents, sir, we have actually, as this is a collaborative project, so for patent, so many
right.
funding is the initial amount is very high. It's 1 lakh or 150 like or so. So that has been paid by the NIST Jodat also. And for us, means for university and colleges, it is impossible to patent because you have to hire a kind of patent lawyer and you have to file the patent.
But what is the director? Some Rao, I think some Rao or some, he is the nice director, no?
Yes, Professor Rao was there at that time, Professor Rao. PG Rao.
I think for them, see, those who are national institute now, so they can have good funding, so they can go for patenting, but not only patenting. I am telling that why only in the TV the advertisement will be there, right? In the home channel only. It should be as a government of India, maybe they can impose the DCGI, Drug Control General, because thousands and thousands of patients, they are suffering from this problem. And in near future, due to this lifestyle, I have good lecture on lifestyle disease also disorders.
Sorry, Professor.
I know, I understand.
And Dr.
Hungry man is angry man. I know.
Also, it is not the matter of hunger also, where the hunger of academics is there.
Yes.
So we can subside our hunger of stomach, right?
But
due to time constraint, right? Due to time constraint, I am very sorry to interfere you.
Welcome, no problem.
Because it is a never-ending discussion.
Yes.
Regarding the polyherbal, again, I am not going to lengthen this discussion. Polyherbal, it's a scientific exploration, patenting. It's a big area of discussion and area of the study. So considering all these things, and especially considering our time constraint, we are very much sorry to interfere in this discussion, though it is very much scientific and interesting discussion. I also like to participate to this discussion, but it will be later on.
Yes.
The time are considered.
Yes. Later all this be normalized.
So I think our committee, I think our committee member will show the certificate and they announce your certificate, all these things. So do this thing, please.
Yes, thank you. Thank you, Professor Rahman and Professor Maiti and Professor Rakesh Kundu. And thank you, everybody, for such a knowledgeable and interactive plenary session. I would now like to share a small token of appreciation for Professor Maiti.
Okay, thank you.
Is my screen visible?
No. Yes. Make it bit enlarged view. It won't be. Anyway. But
yes, it is there.
Thank you so much. So on behalf of Visva-Bharati University and Raidighi College, we would like to express our heartfelt gratitude to Professor Maiti for enriching us with this lecture. And thank you, everybody. Thank you so much, sir.
Thank you, ma'am.
That's the end of this plenary session. Thank you, everybody.
So please send this certificate to him through email.
Yes, sure.
Sir, we will send all the certificates to all speakers. Okay. Thank you very much, sir.
Loco. Yes, Loco,
yes.
You please.
Yeah, I just want to ask one question to our speaker. We have a huge amount of information related to indigenous practices of medicine, but at the same time, we have very little that we have developed into a modern medicine. What could be the reason, and how can we overcome this gap that we are facing today?
This will be a very big discussion.
Yeah, it can be, but in one line I'll tell you.
Yeah.
First of all, our knowledge cannot permit always. Number one. Number two, we have some superficial knowledge.
whatever maybe you believe or not. Number two is that finance is always a challenging job. Getting the volunteers is always a challenging job. Simply, if I ask you are preparing some shampoo in your lab, and if I ask that you use this shampoo on your head first, whether you will do or not, the question will be there. So coming from the indigenous source, we have lots of knowledge. I told you about 4,000 years the knowledge is coming, but there are so many mutations in the knowledge. So that I told you also that sometimes whenever you are going for substantiation, it is not getting the effective result what is reported in the old traditions also. So this is a problem sometimes. But whenever I will visit Visva-Bharati, your zoology department and botany department, I will take one day more so that we can roam and we can sit together, have a nice discussion. Professor Mondal and Professor Hussain is there. I will be there, right? It is my pleasure.
Sorry, Professor Mighty, myself, Habibur Rahman.
Yes.
It's simply Rahman.
Sorry.
Yes. It's okay.
Rahman Sahib. Sorry. Rahman Sahib.
Okay, you are mostly welcome. And you are always regularly coming over to our departments, also our sister department, zoology. So it's not the problem. When the pandemic period will be over, situation will be normal, then definitely you are mostly welcome. You are always welcome to our institute.
Rahman Sahib kache jabo ar ki ache kono osubidha nei.
That's good. Thank you. So with this, I think Madhumita or Anjali, you just help me with this this session. I think now it is the lunch break, I think.
No, sir. We have another speaker.
Oh my God. We are very sorry. Yeah.
90 from Bardhaman University.
Yeah.
Okay, sir.
So thank you.
We'll continue with that.
Yeah.
Thank you very much, sir. Thank you, TK Maiti, sir. Thank you very much.
Now we have arrived at the final plenary lecture. I would like to request Dr. Gyanendra Roth, Department of Botany, Visva-Bharati, and Dr. Ornima Vishwas, Department of Microbiology, Raidighi College, to chair this session. Please, sir. Please, ma'am.
Okay. Am I audible?
Yes, sir.
You are audible.
Okay. Good afternoon to all. And let's welcome to the third plenary lecture. And let me introduce you to the speaker of this plenary lecture, Professor Tushar Kanti Maiti. Sir, welcome to you for this plenary lecture 3. Professor Tushar Kanti Maiti is the professor of botany in the University of Bardhaman. He is having 35 years of experience in research and 20 years of experience in teaching. He acted as a coordinator of a DST FIST program of Department of Botany, Bardhaman University, and also acted as a co-coordinator in microbiology from 2005 to 2008. He's having a lot of awards received, like NC Banerjee Gold Medal for the first in the 3 years of degree programs. Besides, also chair of the Association of Microbiologists in
Hello.
Hello, I think there is some network issue. Dr. Roth, can you hear me?
Just call him, Dr. Roth, just call him.
Can the organizers please call Dr. Roth, or should I take over as the co-chair and introduce Professor Maiti?
Anjulika.
Anjulika, ma'am,
you just call him, the Dr. Roth.
Sir, and just wait, okay.
And inform him that he has been dealing.
I request Dr. Ornima Vishwas, please chair the session. You please introduce our speaker.
Oh, okay. Thank you. So a very good afternoon to all. It is an honor to chair this session along with Dr. Roth. So thank you organizers for giving me this opportunity. I feel honored to introduce Dr. Tushar Kanti Maiti, Professor, Department of Botany, University of DAY-1. It is kind of difficult to describe his long distinguished career in few words or minutes, but I'll try. Professor Maiti has over 20 years of teaching experience in microbiology and over 35 years of research experience. His areas of expertise include plant microbes, PGPR, interaction under stress, diversity of nodule bacteria in legume plants, microbial degradation of pesticides and dibutyl phthalate, microbial production of amino acids, cellulase, and lipase. Professor Maiti has won numerous awards and accolades, including the prestigious N.C. Banerjee Gold Medal from DAY-1 University. He has won PhD fellowships and later postdoctoral research associateships from CSIR and UGC. He acted as fodder botanist and assistant director fodder in ARD Department, Government of West Bengal from 1994 to 2002 and performed research on forage and fodder plant. He also successfully completed training on forage and pasture seed production, grassland management and fodder trees at University of Queensland, Brisbane, Australia as government nominated candidate in the year 1997. From 2002 to 2004, he acted as assistant professor in PG Department of Botany, Darjeeling Government College, West Bengal. Also, he acted as assistant coordinator in microbiology department, BU, from 2005 to 2008. He has also acted as coordinator of DST-FIST project of Department of Botany in BU. He is a life member and the president of the prestigious Association of Microbiologists in India, DAY-1 unit. He is also a life member of the Association of Indian Agricultural Biochemistry. Professor Maiti currently has 6 registered PhD students working under his supervision. 9 students have already been awarded PhD degree and 1 student has been awarded MPhil. He acts as reviewer in Springer and Elsevier journals. He has published 82 research papers in both national and international reputed journals. He has written 6 book chapters and 2 books. He has 30 gene sequences submitted to NCBI and 4 protein models, which he has submitted to PMDB. He has almost 1900 Google Scholar citations with an amazing item index of 46 and H index of 22. So sir, now I welcome you to deliver your talk on role of plant growth promoting rhizobacteria in stress agriculture for sustainable development. I'm sure it's going to be a very enriching experience for us. Welcome, sir.
Yes, I'm very sorry the internet was disconnected and I unable to continue that. Thanks to Dr. Angelica for the continuations.
Thank you. Good afternoon, everybody. Pranam Gurudev, founder of the Visva-Bharati, Honorable Patron, Professor Vidyut Chakraborty, Vice Chancellor, Visva-Bharati, Honorable President and Vice President in this seminar, Professor Tarabhushan Bandyopadhyay, Principal Sikha Bhavan and Visva-Bharati, and Dr. Shashabindu Jana, Principal Raidighi College, respectively. Respected Chairman in this seminar, Professor Sadhir Ranjan Vishwas, Head of the Department of Botany, Visva-Bharati. Respected Joint Convener, Dr. Angelika Rai, Department of Botany, Visva-Bharati, Dr. Mohimita Mojumdar, Head of the Department of Botany, Raidighi College. All respected organizing secretaries, Dr. Buma Dam, Dr. Aninda Vishwas, Department of Botany, Visva-Bharati, Dr. Rikshanani, and Sri Asim Panda, Raidighi College. Respected Chairperson and all respected dignitaries and all learned participants in this 2 days national seminar. It is my pleasure and privilege to be here with you to present a seminar talk in the 2 days national seminar online 2022 on theme Cytogenetics in vitro culture and Phytochemistry of plant and microbe for sustainable use. That is now going on 22nd and 23rd January 2022. And jointly organized by Department of Botany, UCDRS, SAP2 and DST sponsored departments, Sikha Bhavan, Visva-Bharati, Santiniketan, and Department of Botany, Raidighi College, and NAG Accredited College, 24th Pargana South. And many, many thanks to organizing committee in this seminar for inviting me to present a seminar talk. And the topic of my seminar lecture is role of plant growth promoting rhizobacteria in stress agriculture for sustainable development. So due to network problem, I try to stop my video. As the population is increasing rapidly, and it could be crossed 844 billion globally in the year 2030 and 9.1 billion in the year 2050. This problem is more alarming in India because within the year 2025, India would be the most populated country among the world. The production of the food is not increasing significantly, and it needs to be increased to meet up demand on food in the coming year for increasing population explosion. Actually, there are 3 P's problem. 1 P stands for population explosion, and the P for pollution, and last P is for the poverty. The agricultural land cannot be increased. Instead of that is, it is decreasing day by day due to the population explosion and industrialization. The crop production is also affected seriously by various biotic and abiotic stresses in your agriculture system. And if we consider in India, it is actually miniature of the world, which poses the different kind of environmental stresses. It is a vast coastal belt in Eastern Ghat and Western Ghat, many drought-prone areas like the Rajasthan, many flood-prone areas like Gangetic West Bengal or Gangetic Bengal, Gangetic Plain, and the vast cold region in northern part from Kashmir to Meghalaya. Moreover, many biotic stresses, particularly pathogen, lead to decrease the yield of the crop. There are also industrialization is day by day, increasing day by day. Moreover, the sustainable soil fertility is decreased due to the continuous application of the chemical fertilizer, pesticides, which also caused heavy metal toxicity in agricultural land. All are responsible for the decreased agricultural production. And the interesting point is that the implementation of the new varieties of the crops in Green Revolution, we have lost our biodiversity, lost important traditional crops and gene pool, which are resistant to different kinds of the stresses. So in this scenario, PGPR may play an important role. As agricultural production was intensified over the past few decades by management of the biotic and abiotic stresses on crop plants, with increased use of the chemical input or several harmful effects on the environmental quality, to overcome this situation, PGPR play an important role. So what is PGPR? What are PGPR? The microbial communities grown in the rhizosphere, which are beneficial for the plant growth, yield, and crop quality, called the PGPR. And the PGPR may be classified as the biofertilizer, phytostimulator, rhizoremediator, and biopesticides, or the biocontrolling agent. And this PGPR is eco-friendly, inexpensive, effective, and as well as they are sustainable. PGPR facilitates growth and development of plants by several direct and indirect mechanisms. The direct promotion of the plant growth, PGPR facilitates the uptake of nutrients from the environment. And the indirect promotion of the plant growth occurs when PGPR prevents the deleterious effect of the plant pathogen by producing inhibitory substances, or by increasing the natural resistance of the host. So what are the direct mechanisms? These are nitrogen fixation, phosphorus solubilization, sequestration of the iron products by the production of the siderobore, production of the phytohormone, stress controlling and induced systemic tolerance, phytoremediation, enhanced beneficial symbiosis of the host, and ACC deaminase production. I come to later, particularly the last one, the ACC deaminase production. Now, what are the indirect method? The production of the antibiotic and antimicrobial substances, hydrocyanide production of the cell wall degrading enzymes, competitive root colonization, induced systemic resistance, detoxification and degradation of the growth factor, interference of the survival and proliferation of the pathogen. It is not necessary all these direct and indirect mechanisms present in the PGPR. And then no book or no journal clearly mentioned that which character to be present for the called as the PGPR. But the plant growth promoting rhizobacteria, which must be grown in the rhizospheric zone, number one. Number two, they must promote the plant growth by any means. So ACC deaminase is one of the important enzymes produce some PGPR. Actually, ethylene, the important gaseous plant hormone, which have essential beneficial role in the plant. But in any stress condition, it may be the biotic stress or any abiotic stress, it is found that you can see here it is the beneficial peak of ethylene, but in any kind of stress, this ethylene concentration gradually increase. And in the increased ethylene concentration in the plant is so the retarded growth. As for example, their root elongation is minimized. So accumulation or acquisition of the nutrient is minimized. So as a result, the plant is show the stunted growth. So in this scenario, particularly the bacteria, the plant growth promoting rhizobacteria, they synthesize a unique enzyme, that is the ACC deaminase enzyme. This enzyme is exclusively restricted in some microbes, particularly some bacteria and some fungi. So this is actually the Young cycle by which the ethylene is produced. Ethylene is produced from the precursor ACC. And the bacteria is produce the ACC deaminase, and which take the ACC and degrade the ACC by the important enzyme ACC deaminase and produce the ammonia and alpha-ketobutyrate. And this ammonia and alpha-ketobutyrate use the bacteria or use the PGPR as the carbon and nitrogen source. So by which they reduce the concentration or deleterious effect of the ethylene. As the precursor of the ethylene is decreased, so ethylene concentration is increased. So that ACC deaminase reduce any kind of stress. It will be the abiotic stress or any kind of the biotic stress. So the mechanism of the PGR is the threefold. Number one, it's providing the nutrient to the plant by the nitrogen fixation, phytohormone production, solubilization of the phosphate, and protect the plant any kind of the biotic stresses like the siderobore production, ACN production of the antibiotic, production of the fungal cellular degrading enzymes, and also protect the plant from or the ameliorate the abiotic stress by the production of the different kind of the enzymes, particularly oxidase, oxide dismutase, or proline, and production of ACC deaminase, and modulate the levels of the plant stress marker. The agricultural pest is another major biotic stress that reduces the sustainable crop production. Although chemical fertilizer and pesticides have minimized the problem, however, excessive use of this chemical created a tremendous harmful effect on our environment that is ultimately hazardous for the human being. On the other hand, the global demand of the increasing agricultural productivity increases day by day due to the population explosion from the limited land resources has made a challenge for the agricultural scientist. Thus, Both the abiotic and biotic stresses are the major threat to sustainable crop production. So plant scientists are focusing more and more on developing a biosafety and agro-based approach to meet the increasing demand for food by increasing the yield and by lowering the pest invasion and abiotic stress level. In this context, plant growth promoting rhizobacteria are promising one for fulfilling the demand, as they are able to affect directly by improving plant nutrient uptake, modulating growth, stress-related phytohormones, and indirectly by killing the pest and pathogen with antibiotic, hydrolytic enzyme, nutrient limitation process, resulted in increasing crop yield essential for sustainable development. The antagonistic principle of the some PGPR have an important role to control plant pathogen, essentially for eco-friendly management of the pest. Similarly, the elevation of the phytotoxicity of heavy metals or salt by some PGPR can be made by the stress ameliorating ability and respective stress tolerant properties. Moreover, some of them are also able to induce resistance or tolerance, both biotic and abiotic stress of the plant in an eco-friendly manner. So I divided my lecture in the two parts, one is the role of PGPR in biotic stress and role of PGPR in abiotic stress. So first one is the role of PGPR in abiotic stress. So you can see here the mechanism of the biocontrolling action of the plant growth promoting rhizobacteria. In indirect method, they produce antibiotics, lytic enzyme, competition. The antibiotics may be volatile and it may be the non-volatile. So how the PGPR act on the fungus? You can see by the indirect method, they can produce antibiotics. Antibiotics may be the non-volatile or the volatile. In volatile, maybe they produce either HCN, aldehyde, alcohol, ketone, like substances. In non-volatile substances, they produce polyketides like DAPG, heterocyclic nitrogenous compound, phenylpyrrole, cyclic lipopeptides, aminopolyols, and lipopeptides. So different PGI produce different kinds of the chemicals by which they are acting for the fungal cell. Moreover, they also produce some lytic enzymes, some chitinase, cellulase, glucanase, protease, and lipase by which they can degrade the plant, degrade the fungus. And by the direct method, they produce the siderophore by the chelating agent. By the deficiency of the iron, the fungus became killed. Similarly, some nematodes can be killed by some important chemicals by the direct method as well as the indirect method. Viruses also killed by the some PGPR by the antibiosis, and they produce some enzymes, RNase, by which they also kill the viruses. They also kill the insect by the antibiosis, hydrolytic enzyme, toxin. So these are some examples of the different antibiotics produced by some PGPR, effect on the pathogen to protect our crops, the wheat, potato, etc. Similarly, effect on the fungus, chitinase, protease, lipase, and the effect on the bacteria also, effect on the, by which they can degrade the fungal pathogen. In this respect, my laboratory as well as the Visva-Bharati laboratory jointly reported the important bacteria or important PGPR, that is Pseudomonas species, A32 strain, basically the heavy metal resistant strain, but it has the antifungal activity. You can see here different kinds of the antifungal enzyme produced by this bacteria. Another important fungi, that is Pseudomonas P19 strain, isolated from my laboratory actually. And it is basically this bacteria or this PGPR is halotolerant PGPR, isolated from the coastal belt of Odisha. And this bacteria has a rhamnolipid producing bacteria. And this monorhamnomonolipid congener and dirhamnomonolipid congener has exhibited the antifungal activity. You can see here this strain against the Coccobolus myabinus, a rice pathogen. Next, the role of PGPR in abiotic stress. The different kinds of the abiotic stress, the salinity, heat, drought, chilling effect, highlight, heavy metal, and the flood. And every cases, plant growth promoting rhizobacteria has important role by which they ameliorate the abiotic stresses and promote the plant growth. Particularly in India, you can see here the Assam, the drought-prone area, and also Rajasthan is also drought-prone area. North Himalaya, the wheat and moisture. Northeast India, element toxicity, drought, silent stress. Sundarban, heavy metal and salinity. Eastern and Central India, flood, drought, and salinity. In flood condition, oxygen deficiency occurs in the soil, and this oxygen deficiency creates aerobic respiration that supplies a little amount of energy available to transport water from the root to shoot. And fertility rate also decreased or restricted in submerged condition. And plant growth promoted rhizobacteria by this unique mechanism, they ameliorated such kind of stresses. In India, 6.2 million hectares area in rice field are affected by the flooding. And you can see here, the India among the 15 countries, India is the topmost the flood-prone area. The 80% population is exposed on river flood risk worldwide. You can see here the different state of the India where the flood-prone area. Similar number of flood events occurred to the some previous report. And you can see here the crop damage in 2015, about 16,000 crore loss of the crop due to the damage of the crop in 2015. Another important stress or abiotic stress is the drought stress. Drought stress, because the physiological condition of the crop are disrupted, limited water in the soil, decreased crop growth and production. Drought is the major factor limiting crop growth. And in this situation, seed germination growth require sufficient moisture, so seed development is either slowed or altogether stopped. So in this scenario, plant growth promoting rhizobacteria by some important direct mechanism alleviate the drought stress and promote the plant growth. The most important stress is the salt stress. About 77% of the world's total arable land affected by the salt. And in the coming 25 years, it may reach up to 30% of the total cultivated land. The extent of soil salinity of up to 50% of total agricultural land will be lost due to the salinity and sodicity by 2050. How alarming you can see. About 7 million hectares of the land is affected by the salt stress in India. These agricultural lands are usually not fit for the crop production as high concentration of the soluble salts cause poor soil physiological and biochemical properties. And in this scenario, rhizobacterial microorganisms play a key role in modulating cellular response and enabling plant tolerant to all types of such stresses. You can see in worldwide scenario, the salt affected area in the world, and about 27.20% of saline and sodic soil is found in Asian, Pacific, and Australia. Similarly, saline and sodic soil. Salt affected soil in India, you can see the West Bengal, Odisha, Andhra Pradesh, as well as the Gujarat. Extent and distribution of soil affected soil in India. How alarming this. In our district, the soil affected district in the India. And what is the reason behind it? Two causes behind the soil salinity and natural and anthropogenic activities. Natural causes due to the climate, location, hydrology, and geomorphology. An anthropogenic factor includes excessive irrigation, inadequate soil topography, inadequate drainage, and saline irrigation chemical abuse. So these are the main cause of the salinity. Salinity mainly decreased soil physiology, increased soil compactness, decrease the conductivity of the soil, also make the nutrient and oxygen deficiency in root, reduce seed germination, seed growth, flowering, fruiting, nitrogen activity in the symbiotic bacteria. And you can see here the effect of salinity in the plant, the osmotic effect of photosynthesis, leaf senescence, inhibition root growth and cell elongation, inhibits enzyme activity, inhibit protein synthesis, inhibits seed germination, affect nutrient uptake. So these are the impact of salinity on crop plants. And in the saline condition, plant, particularly rice plant, have some important genes. And by the application of the PGPR, these particular genes are upregulated and make the plant saline tolerance. So in this context, we have isolated some important halo-tolerant PGPR strain that are also found to increase plant growth under saline condition. So we have isolated some important PGPR strain from the coastal belt of Odisha. P12, P13, P15, P19, P23. In this way, we have isolated some important PGPR strain, and they possess the important PGPR activity also, or PGPR trait. Among we have selected some PGPR strain, among which P23 strain, which is identified as introvector by the modern approach of bacterial taxonomy, that is by the multi-top, by the morphological character, by the DNA sequence homology, identified as introvector species, the P23 strain. And another strain is P50 strain, that is also isolated from the Odisha, coastal belt of the Odisha, and it is also identified as Burkholderia. And we use these two strain under the salt condition to the rice seed germination. Two rice cultivar we have chosen here. Number one is the Ratna. We apply the P23 strain. And another Sorno Masuri rice cultivar, we applied the P50 strain. And observe that EC50 of these two is 150 millimolar and 185 millimolar respectively. And if we application of the PGPR P23 strain, that is the introvector, the germination is increased, more or less normal condition. Similarly, application of the P50 strain, that is the Bartholleria, the seed germination percentage also increased under the saline condition. Application of P23 strain in the salt condition, we have observed that root face weight, root dry weight, and root length. This is here the control condition, that is the root face weight and the root dry weight. But after the application of the salt, decrease the root face weight and the root dry weight. But application of the saline and PGPR, the root face weight and root dry weight is also increased. Here you also see the root length also. Similarly, root face weight and root dry weight also increased, and chlorophyll content is also increased. By the application of this ACC-D-amine containing PGPR, the morphological effect also increase, as well as some antioxidant enzymes also regulated by this PGR to ameliorate this salt stress. Same thing also happens in case of the Bartholleria. By application of the Bartholleria, seed germination percentage, root shoot weight, length, amylase, protease, and oxygen content and chlorophyll content is also appreciably increased by the application of this PGPR under the salt stress condition. Antioxidant enzymes like the SOD, catalase, MDA, proline, total soluble sugar, and total protein is also increased by the application of the salt-tolerant Bartholleria species. So we can conclude that PGPR with ACC-deaminase lowers the ethylene production, reduces the ROS, production of the osmoprotectant like the proline. They also produce the extracellular polysaccharide by decrease the translocation of the sodium ion. They produce the phytohormone and produce the some importance metabolite by which they increase the fertility. So in this way, halo-tolerant PGPR ameliorate salt stress. So this is some example of this halo-tolerant PGPR. Another important stress is the heavy metal stress. No question of what are heavy metals. Heavy metals are the metal with a density above 5 grams per cubic centimeter and a specific gravity at least 5 times higher than water. And some metalloids also. The metalloid means any chemical element that has the properties in between those of the metal and non-metal, and which is also called the semi-metal. And we have restricted our particularly our research mainly the arsenic, cadmium, and some lead. Most of the heavy metals are dangerously toxic. These are the some effect of the heavy metal on human being. You can see here cadmium poisoning, arsenic poisoning, lead poisoning, and mercury poisoning. And there is the threshold limit of heavy metal in the agricultural soil. So with the cadmium, you can see here 0.176. So in this way, there is some threshold limit, and excess of this condition they can accumulate in the grain and which are biomagnified in our body. Here the toxic effect of the heavy metal on the plant, to damage the DNA, damage the protein. But some heavy metal tolerant bacteria, particularly PGPR, they can accumulate by bioabsorption or bioleaching, biotransformation, biomineralization, by which they resist the heavy metal. So these are the different sources of the heavy metal contamination, among which the important one is the chemical fertilizer. Arsenic, one of the heavy actual metalloid, which it accumulates ultimately in our brain. Similarly, the cadmium, which also are accumulated in the grain. So in this context, we have also isolated some heavy metal and metalloid resistant plant growth promoted rhizobacteria. These strains are found to improve plant growth and reduce heavy metals and metalloids in plants. These strains can be used for the effective recognition of the metal contaminated agriculture field and in enhanced crop production. So we have isolated some important heavy metal resistant bacteria from the Durgapur alloy steel plant, iron steel plant. Actually, the leaching or these heavy metal industrial waste come into the agricultural soil, from which we have isolated this kind of the bacteria. And among these, we have isolated particularly steam, and we first check the soil profile, among which we observe that the cadmium, lead, and arsenic concentration is very, very high in this pore site. And we isolated this kind of bacteria. These are the heavy metal tolerant bacteria. Peak or minimum inhibitor concentration we have shown here. K2, K4, K5, K6, and K32 strain. All are resistant up to 4,000 microgram per ml of the cadmium, and about 2,000 to 3,000 microgram of the lead, and 100 to 2,000 microgram per ml of the arsenic. We have selected one of the strain and identified by the modern approach of bacterial taxonomy as Klebsiella pneumoniae. Another bacteria is identified as the Enterobacter erogens. These bacteria are identified by both the morphological character, hemanalysis, multi-tube analysis, and the modern approach of bacterial taxonomy, that is the 16 sequence homology. Another bacteria we have identified as the Bacillus argyvohti, and we have isolated AS4, AS6, AS7, AS11, AS12, AS14. They are the 6 arsenic-resistant bacteria, among which the AS6 we have identified as the Bacillus argyvohti by the modern approach of bacterial taxonomy. The same procedure. Interestingly, these bacteria are, particularly the Klebsiella pneumoniae, we have found that they can ameliorate maximum amount of the heavy metal, or particularly the cadmium here, and reduce the cadmium concentration of the 98% cadmium removal ability of this particular bacterial strain. And at the same time, it was found that they can accumulate cadmium their outer wall, the bioabsorbent. By the same time, and EDX analysis, we have found that they can ameliorate the cadmium. Similarly, Enterobacter erogens, another bacteria, they also remove the cadmium from the broth, but they can accumulate the cadmium within their cell. You can see here by the TAME analysis, you can see that they can accumulate cadmium within the bacterial cell. Both the same TAME analysis, it is observed that you can, and EDX analysis, you observe that they also accumulate cadmium. You can see here cadmium, but here the cadmium is absent in the control condition. Similarly, here the cadmium is present, but in the control condition there is no cadmium. But by this graph, you can see they can accumulate the cadmium from the broth. Another bacteria, there is the Bacillus arjobhatti. By the FT analysis, by the XRF and XRD, clearly found that they can absorb the cadmium, and by the TAME analysis and SAME analysis also observed that they accumulate their arsenic, particular arsenic, in their outer cell wall. So the different mechanism of the heavy metal amelioration by the different bacteria. In this slide, we try to know how the different phytohormone production or nitrogen fixation or the soluble phosphate can be affected in the higher concentration of the cadmium. So here we gradually increase the cadmium concentration and the IF production is gradually decreased. Similarly, nitrogen fixation is also decreased. But you can see the 3000 microgram per ml, such kind of concentration is not available in the soil. Similarly, here in this slide, we can see here the germination percentage also decreased by the seed of the rice, but application of the PGPA, particularly this bacteria by the Enterobacter, the germination percentage increased. So morphological character, seedling vigor index, root weight, shoot weight, root trace weight, shoot trace weight, root dry weight, shoot dry weight also increased by the application of the PGPA. In this slide, we also show that some important enzymes activity and some antioxidant enzymes activity is also ameliorated by the bacterial strain. So by this way, Enterobacter erosions alleviate the phytotoxic effect of the cadmium and promote the rice seedling growth. In conclusion, we can say that application of the bacteria, plant growth promotion, cadmium removal efficiency by accumulation. So in the normal condition or the heavy metal contaminated condition, the growth of the rice seedling is stunted, but the application of the PGPA, you can see the growth of the plant or growth of the seedling can be increased. So in summary, we can say the selected heavy metal resistant strain and salt tolerant PGPA strains are successfully alleviate heavy metal and salt stress in rice seedling by reducing various stress-related harmful effects of the plant. Therefore, these strains may be useful for sustainable agriculture in metal containers or the coastal rice field under the moderate saline condition after field trial. In conclusion, we may say that as a result, use of such PGPL may be a viable alternative to chemical fertilizer and biocontrolling agent, having also stress removing abilities in certain negative effect of the stresses. A lot of studies showed that inoculation with PGPR resulted in significant increase in the growth enhancement, including seed germination, root and shoot growth, plant weight, resulting higher yield of the different crops, and suppression of the plant disease, insect and nematodes. Thus, PGPR are now the potential tools for sustainable agriculture and promising trend for the future. However, every PGPR strains have the limitation in terms of their specific metal tolerance or salt tolerance level, acclimatization of the specific environmental condition, plant specification for root colonization, susceptibility of the soil-borne pathogen attack, level of plant growth promoting activity under the both biotic and abiotic stress condition. Therefore, global demand of the isolation of the more and more stress-tolerant PGPR strain is currently a very emerging area of research to apply in the heavy metal contaminated field or any kind of stress-related issues for effective reclamation and to reduce the health risk of the human being. But one important information I would like to share that the application of the PGPR, by application of this reported PGPR, is not so effective in the field condition. This is due to the natural soil microbe. So, indigenous PGPR strain is more effective, number one. Number two, the application of the PGPR or method application is very, very important. So lastly, the acknowledgement. CSR and DST is gratefully acknowledged for the financial support. Different authors who are reported the PGPR activity in the different journals. And lastly, my research scholars are the key players who did these works. So thank you. Thank you for your patience sharing.
Thank you so much, Professor Maiti. Professor Roth, will you please take over?
Yes, thank you, sir, for your enlightening lecture. We are really very happy to know. I'm personally, because I works also on this line of stress, and I'm really very happy that you have done such a tremendous work in all this stress, and you have such a nice publications you have produced. And I'm quite sure that all the participants are benefited with this lecture. And though we are now a little late, so if the participants have any questions, maybe one or two, can come forward, can unmute themselves and can ask. Very short questions if you have.
Good afternoon, Tushar. I'm Bumba.
Okay, thank you.
Actually, I would like one clarification from you regarding this use of halophilic bacteria. Right. So, is there any report where this bacteria actually help by reducing the amount of salt? I mean, those plant growth promoting activities are fine, but is there any report that where bacteria have helped by reducing the amount of salt? Or is it possible for that matter?
Actually, I have not received any kind of information, but one thing is that the bacteria has produced extracellular polysaccharide, EPS, by which they can absorb the salt. And
that's what I'm asking, whether this strategy has been used or whether it is feasible or not.
Yes.
That's what I want to know.
Yes, this strategy is also used.
Okay.
Sir, I have a clarification I want. So whether these bacteria work in multiple stress conditions, so you individually see that in this drought or in the heavy metals. So whether, because in the field conditions, we have multiple stresses. So have you studied some of these things?
Regarding the multiple stresses, it may be the multiple abiotic stress or the biotic and abiotic. So we have worked on the particularly, I've shown the slide, particularly a Pseudomonas bacteria or Pseudomonas pgps strain, which have the particularly heavy metal resistant strain. That is, it can able to tolerate up to 3000 microgram per ml of the heavy metal of the cadmium. And as well as the heavy metal, not only the cadmium, but also it is also tolerated the cadmium, lead and arsenic. But this particular strain also has ability to biocontrol, act as a biocontrolling agent. Clear? So in that scenario, it is occurred that the multiple resistant, that means in any soil, the individual heavy metal is not not avail this condition. Heavy metal condition must be the all kind of heavy metal may be present there. So in that scenario, the particular this we have isolated, we have reported that the Pseudomonas strain has ability not only to resistant to heavy metal, but also it is also biocontrolling agent also. It is some more antifungal activity. Clear?
Okay.
And another important bacteria we have also not I have not shown here. Some bacteria, particularly Pantua, a important bacteria, Pantua, which is resistant to arsenic as well as salt, both.
Okay, yeah, that's great. Any other queries from the participants? Okay, so Dr. Arunima Biswa, so can you just do the felicitation thing?
Yes, sir. Thank you. Thank you, sir. So Professor Maiti, once again, thanks a lot. That was indeed a very informative, lucid, and really outstanding presentation on a very essential topic. And you've also shared your lab work with us and your publications. Your lab has done extensive, outstanding work on PGPR, especially with respect to metal contamination and salinity stresses. Thanks for sharing them with us. And on our behalf, here is a small token of appreciation. I'd like to say, is it visible?
Thank you. Yes, it is visible. Thank you for your night.
Thank you, sir. Thank you once again. And thank you, Professor Roth.
Yeah, thank you, sir.
Thank you, sir. Thank you, ma'am. So today's plenary session is over now. Our respective sessions for oral and poster presentations has already been initiated. All of you are requested to join the respective oral and poster presentation following provided Google Meet links. We will meet again tomorrow at 10 a.m.
Thank you all.
Thank you very much. On behalf of organizing committee, I extend my gratitude and thank you very much. We will join tomorrow. So today's session ends here, and I am also thankful to Devoshmita and Kunal for conducting this session.